Good evening, everyone. We're already a little behind schedule, so um, we'll know people will come in over time. But I wanted to get started by making a few announcements. First, about tomorrow, we got together and we decided that what we're going to do is at 9 o'clock, you'll go up to the second floor. There's 3 rooms. They're labeled with the 3 of the morning themes. Pick one and go in. We have a coffee break from 10:15 to 10:45. That'll be back down here. And then we thought it might be good if when everyone went back up after coffee break, pick a different session, stir it up a little, and add your 2 cents to 2 different topics. Okay. Um, Second thing I wanted to mention is that, and here someone has to hear that. This afternoon, one person of the 10 people who had signed up for a tour managed to actually remember to go on the tour. And he said it was great. So the people who are running it said that those of you who missed today and want to do it, show up tomorrow and just do it. Okay. 12:45 right out front here. Okay. So you get a second chance on this. And then the third announcement here is I'd like to take all of the students out to lunch tomorrow. So when we wrap up here around noon, we'll walk downtown, and depending on how rainy it is, we've got a couple potential options there. Okay, so that's. Do I have the fourth announcement? I don't. Yeah. Okay. So let's start with the evening session. We'll just go a little late and squeeze the coffee break a bit. We're going to start tonight with a presentation by Dr. Jebri. She's joining us remotely from the UK, and she'll be talking about, well, you can read it, Agulhai's current influence on productive upwelling regimes revealed by machine learning.
Should I start? Sorry. Okay, cool. Hi, everyone. Thank you very much for having me. So my name is Fatma Jebri, and I'm a satellite oceanographer at the National Oceanography Center in Southampton. So the work I'm about to present was conducted in the framework of Solstice, which is a 4-year collaborative project with societal and economic impact in the Western Indian Ocean. What you are about to see more exactly is how machine learning can help us study the influence of the Agulhas current and productive upwelling regimes on the Agulhas bank. So I'm going to begin with the overall context of the study and its key question. Second, I'll explain our approach. And finally, I'll take you through the main results and conclusions. So in the Western Indian Ocean and the Agulhas Bank in particular, a seasonal upwelling known as the cold ridge, so highlighted here with the green shaded area on the left, it takes place on the Agulhas Bank from November to March when the northwestward winds blow parallel to the south coast. And these productive waters help sustain rich ecosystems and fishing activities, which are important for coastal fishing communities. This region is also characterized by complex dynamic, as you can see on the left here. So it's dominated by the Agulhas current, which affects and is influenced by the hydrography of the Agulhas bank. So we have current rings, eddies, filaments, among other processes. However, it is unclear what are the typical patterns of currents which influence the phytoplankton availability during the upwelling season and how they vary. So that's what we will try to answer. So to answer this question, we used machine learning and satellite observation, which offer us new ways for addressing the quite unique challenges that ocean physical and biological interaction pose, especially in complex regions with non-linear processes, such as the Agulhas region. So you probably already know what machine learning is about, but I am still going to give a quick overview as it will help me introduce the method we used. So in very simple terms, machine learning is a subset of AI, where algorithm performs tasks without explicit programming. Machine learning algorithms can be categorized into three main classes. So we have supervised learning, which is task-driven. We have unsupervised learning, which is data-driven, and reinforcement learning. And this categorizations applies also to deep learning algorithm, which include recurrent neural network and self-organizing maps. How deep learning works is that it uses an artificial structure holding a neural network composed of neurons in a number of layers. So some neurons will receive input values, and so these are the values that are known to us and that we want to analyze. And that's called the input layer. And then we have the output value that we would like to determine, and that's the output layer. And in between, we have a number of hidden layers where neuron synapses are formed. So that's a very basic view of neural networks. So here, what we have done is that we applied an unsupervised deep learning approach, the neural clustering self-organizing maps, called also SOM, to 24 years of satellite currents, sea surface temperature, and chlorophyll A over the period November to March 1997-2020. So the functioning of the SOM is summarized in this chart. I know it might seem like a lot to take in one go, but it's more straightforward than it seems. So if we remember the structure of a neural network from the previous slide, it has two important layers. So we have the input layer and the output layer. For the SOM algorithm, the input layer will take the three-dimensional satellite data concatenated into a row vector. And the output layer is composed of a two-dimensional map. So here the size is 3 by 4, so leading to 12 neurons. And each neuron is modified through the iterative training process called self-organizing. And when a neuron is found, its weight vectors and neighboring neurons are also updated, and so on until the algorithms converge. Now, from the SOM patterns extraction, what we obtained are 12 characteristic sea surface temperature and surface currents pattern over our study area during November to March 1997-2020. So here, please note that the superimposed currents, meaning the thick colored arrows, are just added here. They are not a direct result from the algorithm. We added them for better clarity on the flow behavior. And the percentage that you see between parentheses represent the frequency of occurrence of each pattern during the 24 years of observation. So if we look more closely to this output and we focus on the main Agulhas current configuration, so indicated with the thick black arrows, these patterns can be grouped into 4 AC modes. So we have an AC flowing along southwestward along the topography. So those would be patterns 1 to 4, 7 to 8, and 10 to 11. And this is the most frequent mode. We have also an AC with a cyclonic meander near shelf mode. So that would be pattern 9 and 12. And this is the second most frequent mode. And we have the AC early reflection, so seen in pattern 5, which is the least frequent. And finally, we have the AC with a cyclonic meander off shelf mode, which is seen in pattern 6 and is also rare. These SOM outputs return also 12 characteristic chlorophyll A patterns, which relate to the sea surface temperature and current patterns that we've seen in the previous slide. From the 12 characteristic chlorophyll A patterns, we can deduce that productive cold ridge upwelling is found in the AC early retroflexion mode, so pattern 5 here. Also the AC with a cyclonic meander near shelf mode, so pattern 9 and 12. And half of the AC along the shelf edge patterns. These more productive patterns can generally be associated with a strong southwestward flow over the central bank. So as that we can see here in pattern 12, so symbolized by the thick pink arrows. And this can be caused by the intrusion of the AC from the east bank or via an cyclonic meander, such as in pattern 5 here, as a result from the earlier reflection. Now, when it comes to less pronounced Coleridge upwelling, they can be seen in the AC with a cyclonic meander off shelf mode, so pattern 6 here, and in the remaining patterns of the AC along the shelf edge mode, so pattern 1 and 4 and 10 to 11. Now, if we want to see to what type of current flow these less pronounced less pronounced Coleridge upwelling are associated with, they can be linked to a weaker southwest flow over the central bank, as can be seen here in pattern 6, or a strong northwest flow on the east bank, such as in pattern 1 and 4, or it can be also linked to a stronger northwest flow on the central bank, such as in pattern 11 and 10, and these might hinder the extension of the Coleridge further to the southwest. The sum enables also to look at the interannual variation of the extracted pattern, meaning the Winnan neurons' variations with time. So their time series show here a strong year-to-year variability from 1997 to 2020. And from this time series, what we can see is a typical pattern sequence, such as exemplified here in 2016-17, so highlighted with the light blue area. And if we zoom in, we can see the exact patterns changes during that period. We can also distinguish anomalous events compared to the shape of the rest of the time series, so highlighted here with the gray shaded area. So we have 2000, 2001, 2005, 2006, and 2010-11. But note that there are also other events that are also quite irregular. But next, I'm just going to focus on more on the highlighted events. So in typical conditions, as exemplified by November to March 2016-17, only the AC along the shelf edge mode is active. So we have pattern 1 to 4, 7 to 8, and 10 to 11. And note here that these brown arrows represent the pattern's trajectory as they change with time during November to March 2016-17. So basically, it's just a better visual of the zoom in time series of the pattern during that period. Now, in terms of productivity, the typical condition show patterns that oscillate with about 50-50 chance between less productive and more productive Coleridge upwellings. And when it comes to anomalous patterns of sequences, as exemplified here by November to March 2000-2001, they show usually less frequent and a fewer number of patterns than a typical season. So here we have only 4 patterns. Also, what was particular about November to March 2000-2001 event, it started with pattern 6 instead of pattern 3, like in a typical season. And the earlier reflection in pattern 5 here was active from December 2000 to January 2001, according to our analysis. And this was previously linked in the literature to the delayed effect of the 1997-1998 Super El Nino and extreme positive IOD on the Agulhas retroflexion and the ring shedding. And the anticyclonic meander that detached from the main current due to this extreme early reflection, it contributed to the strong southwestward flow. So extended Coleridge over the central bank, and hence more widespread productivity. So that productivity in the 2000-2001 upwelling season was overall high, as the productive pattern were most frequent. So we have pattern 5, also 9 and 12, and together they represent about 82% of the occurrences. Another example of anomalous pattern sequence is November to March 2010-11 event. And although it starts quite like a regular sequence, so we have pattern 3 to 1, but it isolated quite a lot between them. And also it shows the rare AC with early retroflexion mode, so pattern 5. And also we have the AC with the cyclonic meander near shelf edge mode in pattern 9 and 12. And 4 patterns of the AC along the shelf edge mode that were also active. So the 2010-11 event was mostly productive, with about 78% of the realization representing more productive pattern. And also 2010-11 event coincided with the second most intense La Nina on record since 1979. And we know from the literature that during La Nina, wind-driven coastal upwelling enhanced phytoplankton growth is favored over the bank. And this agrees more or less with the mostly productive nature of 2010-11 event that was found from our analysis here. So this again gave us more confidence in the method that it's actually picking up realistic dynamics. Now, to conclude, I'd like to go over the main findings. So first, we've seen that the 12 extracted SOM patterns showed 4 modes of the AC system. So we have the AC along the topography, flowing southwestward. We have the AC with the cyclonic meander near shelf edge mode, the AC with the cyclonic meander off shelf mode, and the AC early retroflexion. And these modes influence the circulation and the phytoplankton productivity on the shelf. We also identified the types of flow that correspond to productive upwelling regimes and which are responsible for reduced levels of productivity. We looked at the interannual variability of the retrieved patterns and how the typical conditions are usually composed of the AC along the shelf edge mode only, while anomalous conditions show fewer number of patterns and patterns that are usually less frequent. Also, the high-low productivity of these events seem to be linked to the occurrence of extreme phases and climate variability modes, such as ENSO or IOD. And finally, the novel aspect of the study lies in the application of the SOM to extract spatial patterns that relates ocean physics and biological processes. And this machine learning analysis can also be used with other types of data, model data, sorry, or extended to other oceanic regions. Sorry. So as a next step, we aim to integrate into the SOM other variables such as the winds to account for atmospheric effects and study links with the biological response. And that's it for me. More details can be found in our paper if you'd like to check that out. And thank you for listening. And I'd love to have any questions.
Thank you. Do we have any questions here from the audience?
Hello, thank you. Very interesting talk. So the question is, what are the drivers of these variability patterns?
Okay, so the main driver of the nutrient supply, as we understand it, so it's a seasonal upwelling. It's mainly caused by the northwestward winds as they blow parallel to the south coast between November and March each year. So they cause a pattern that was named in the literature as the cold ridge. It was first detected around 1998, and then later on with Roberts and al. 2005 work, it was better visualized from satellite data. So we have a cold filament of nutrient-rich water that flow along more or less follow more or less the 100-meter isobath from the Titicaca coast, flowing southwestward along the shelf. So the main driver is the wind, but what we try to understand here is the effect of the current flow on the advective effect of the current flow on this phytoplankton availability that was common from the wind-driven upwelling.
All right, thank you. Since we are already running somewhat behind, I'd just leave it with one question for now. We have her, Dr. Jebre's address, and you can address additional questions to her at your leisure. But right now, we're going to switch over to Dr. Adam Greer, who is going to, what are we going to talk about? Adam's talk title is Resolving the Influence of Water Column Structure on Production and Mesozooplankton Traits in the South Atlantic Plate.
All right, can everyone, I guess, yeah, I can hear myself. All right, well, thank you everyone for being here this evening. I'm going to be talking about some new results out of our lab group using imaging systems like the one pictured here. It makes a lot of really pretty pictures of all the life in these western boundary currents on the shelf in particular. And it's actually helping us understand a lot about the fine scale responses of these organisms to the key processes in these systems. So I'm going to be talking about the Gulf Stream, which probably needs no introduction at this point, but it is a western boundary current that has this interesting feature where it goes from a relatively narrow shelf to a much wider shelf. And from a 2D MODIS satellite image, it looks quite simple, but there's a lot of interesting processes happening below that we've talked about today. But one of the key processes is that in addition to these meanders of the Gulf Stream itself, you get a wind regime kind of in the mid-shelf region. This is between 20 meters to 45 or 50 meters isobaths. And when that wind is blowing a certain direction, you get upwelling of cold, nutrient-rich water onto the shelf. And if you were to take a CTD and profile it through one of these upwelling events, you would get a very highly structured water column vertically. And so this is a really key process for driving stratification, in particular in the summertime. And because this is a wide shelf up off the coast of Georgia, you get these longer residence times, which allows the biology to respond. And so we can really expect to learn something quite fundamental about multi-trophic level responses on shelf ecosystems in response to these upwelling events. And so the zooplankton are animals that occupy this intermediate trophic level between the primary producers and the higher trophic level fisheries. And as we all know, the primary producers start with these phytoplankton. In this case, this is mostly different types of diatoms that form these chains. And these are some of the most abundant plankton on the shelf in the South Atlantic Bight. And the traditional or conventional food web that we know a lot about is that these phytoplankton are consumed by small crustaceans, these copepods, and that's really good food for the higher trophic level fisheries, such as forage fish like this anchovy. And these copepods produce fecal pellets that sink rather rapidly, but they're not the only grazers out there. There's also things like this. This is a pelagic tunicate known as a doliolid, and it's gelatinous, it's relatively large, it's low in carbon content, and so it likely has a different suite of predators, including a lot of different gelatinous predators out there. In addition, the fecal pellets from the doliolid sink much more slowly, and so you might get much more colonization of the microbial food web and then ramping up of a lot of those microbial processes. So when you take a big look at this, you can kind of see that these intermediate trophic levels really influence what the rest of the food web will look like. And so the overarching question of some of our work currently is which pathway is favored and why? Are there certain conditions that will tip the scales in favor of copepods versus something more gelatinous? And to study this, we've been using a towed shadowgraph imaging system. It looks like this on the left here. And the way it works is that you have a light in one pod and a camera in the other pod. So that's happening right here. And the LED goes through a very small pinhole, and that hits a plano-convex lens that makes all the light beams go parallel to one another. This is collimated light. It gets refocused into the camera. And so everything in this imaging volume remains in focus, and that allows you to look at a relatively large volume of water. And so we can get a pretty good understanding of some of these larger zooplankton out there. We tow this system behind a ship, and it's integrated with a bunch of different oceanographic sensors like a CTD, dissolved oxygen, chlorophyll A fluorescence. And it produces a really kind of exciting data set where you have each individual planktonic animal has a specific depth where it was captured and all the different oceanographic data associated with that particular parcel. And in addition to kind of giving you a really good understanding of a lot of these fragile gelatinous organisms, there are many other transparent organisms that are kind of hard to image that show up really well in shadowgraph form. So this is a leptocephalus larva. This is an eel, a larval eel. You can barely see it, but when you put it in shadowgraph, it really pops. It's really easy to design software that will segment that or extract it, and then we can actually learn a lot from the images. And so the system we've been using is called the Modular Deep Focus Plankton Imager. It's derived from the ISIS, which is a system I've used for a really long time. This one is modular, meaning it's easier to deploy in different situations, particularly for station-based oceanography. And as you can see, it's kind of gone through various iterations of the vehicle, from something simpler and smaller to a little bit larger and heavier. And we've been towing it in addition to, we've been using a fully vertically integrating net. And if we look at the data that is generated by the MDPI compared to a net, there's some pretty striking differences. The white bars are showing the data from the MDPI. These are 1 meter bends, and this vertical line is showing what is detected with the vertically integrating net. And so what you can see is that doliolids, which are these fragile gelatinous organisms, they're really underestimated by the net. Shrimp is a little much more similar, but a little high in the net for some reason. But when you break it down, the doliolids are about 3 times higher in the MDPI compared to the net system. And so you're really getting kind of an accurate representation of this, what I like to call the fragile food web, all these organisms out there that are really hard to sample because they're just not, they're basically destroyed by these net systems. And so this is kind of what it looks like when we're towing it. You get the vehicle itself, the information is going through this thin yellow fiber optic cable that goes all the way up to the ship, where we get a live feed of all the data coming through. We can see the sensors, we can see a live feed of the images. It's pretty exciting when you go through kind of interesting features in the water column. And the nice thing is that all the sensors are independent of one another. So if you have a serial feed for the sensor, you can record those independently. And we tow it about 2 to 3 knots. We go 4 to 6 times up and down at a given station. And so we've been sampling with this system in the summer of 2021, but we've actually sampled a lot longer, but I'm going to be talking about summer in particular today. 24 stations at the mid-shelf, so between 25 and 45 meters deep, from Savannah down to Cape Canaveral, Florida. We process the images automatically by cutting out essentially organisms or particles above a certain size class. And I also want to draw your attention to Patrick Duffy's poster, which is out there. And he really looks at the broader time series that we've collected throughout the year, so you can see the whole year-long variability. But I'm just going to be focusing on summer today. And so here are some of the data that we've been getting. This is showing where we sampled, and the size of the dot shows how many particles or plankton were in that particular tow. And this is averaged throughout the water column. And the shape of the dot is actually showing whether the vertical profile was determined to be sharp or smooth. And how we did that was we used the buoyancy frequency, and we just calculated average temperature in this case, but it was based on density. And you can see a pretty strong, basically a nice difference between these ones that we call smooth, where there isn't some buoyancy frequency threshold that's eclipsed. And so we consider these from 9 and 14 on the map to be sharply stratified. And so we divided that up into essentially 3 habitats. So comparing what it's like for zooplankton in the smooth stratified versus the sharply stratified. And so the below area, as you can probably imagine, is really high in chlorophyll A, because that's when there's intrusion of that deep nutrient-rich water onto the shelf. And so for our analysis, these are a bunch of examples of organisms from the South Atlantic Bight. Each one, you can look at it and tell a story about what it's doing, what its species, if you want to. But we really wanted to focus on the traits of the organisms. So what are they actually doing in the environment, rather than going into the details of what exact taxonomic category we could assign. So these are like larval crab, this is a larval stomatopod. We got a bunch of different, this is a larval lobster. So there's a lot of stuff there. And if we break it down by taxa into broad taxonomic groups, this is what an average water column looks like. You can see this orange bar is the number of hydromedusae. So these jellies are really, really common all throughout the South Atlantic Bight. But we're not really getting at the function. And so that's why we want to look at the traits and group a lot of these different taxa together to figure out what they're actually doing in the environment. And we can actually see those changes in function on a very fine scale with these imaging systems. And so we focused on 4 key traits, the first one being size, which is kind of the classic trait. It's something that is really easy to measure, and it's often used in a whole bunch of different studies. But we also looked at mobility, so if the organism was able to move rapidly, or if it tended to stay in one place, or didn't have very good swimming ability. Feeding strategy as well, so how they capture prey can indicate what they would target. And then also the carbon content. So low carbon content organisms are gelatinous. They have low carbon density versus something like a crustacean or a copepod, very high carbon density. And so we break these down by those habitats that I mentioned. So above, and then pycnocline below, and then smooth is that pink color. This is based on size. And so you have 3 size classes. There's not really that big of a difference between the sizes. But when we start looking at the other traits, we notice some pretty strong differences. For instance, with regards to motility, there's a lot more passive swimming animals in that smoothly stratified water column. And you start to see sufficient and quick or fast swimming animals being more abundant in the pycnocline and below. And similarly, passive ambush feeders are well represented in that smooth water column, and you see more filter feeders in the pycnocline and above in the very sharply stratified water columns. Carbon content as well, a lot of low carbon animals and then high carbon content animals tend to be below in that high chlorophyll water as well. And if we look at the diversity in terms of Pailu's evenness, so if it's a higher value, it just means there's more diversity in terms of the trait, and if it's towards 0, it means there's less. And so what we're seeing here is that the smoothly stratified water columns in the purple tend to have lower trait diversity, and especially in terms of carbon and their trophic or their feeding mode, but for size, it doesn't really see much of a difference. And so sharply stratified stations are more diverse, they tend to be, and this suggests kind of a sequence of events for intrusions onto the shelf where you may get a lot of different habitat types promoting a lot of trait diversity, and then as that starts to be mixed out, you start to see increasing dominance of these gelatinous, passive, low-carbon animals. But the caveat is that most of our measurements were taken during the day, so we do have a few night samples where we can actually look at how those distributions may change day or night. And so one of the great things about this project we've been working on for about a year and a half is that we've actually had the opportunity to go back to the same places in different years, in addition to sampling every 3 weeks for almost an entire year. And so in the data I talked about in 2021, the minimum at the bottom of the those temperature profiles was around 20 degrees Celsius. But we went back the next year in 2022, the temperature was much colder at the bottom, so it suggests a recent upwelling event. And this deep water was dominated by these really large protists called Colodaria. They're radiolarians that are mixotrophic. They fluoresce like crazy. And so what I'm going to show you, and there are also a lot of doliolids above the recently upwelled water. And so what I'm going to show you here is an actual video from one of these profiles. The red dot is showing you where you are in the water column. So what here, this is temperature and chlorophyll A as a relative measure of fluorescence. And so this red dot is going to go down, and you'll see kind of how the water column changes at this transition zone. And in addition, we had a GoPro on the vehicle itself. And so I'm going to show you actually what it would look like if you were scuba diving, and then also what it looks like in shadowgraph vision. So let me just play this. And so I kind of isolated it so that there's a doliolid. So you can see there's pretty abundant right above this transition zone. And then as we go down, you can kind of keep an eye on the right top screen, and you'll start to see it kind of get greener, and it almost looks like the vehicle is about to hit the bottom, but it's just this huge aggregation of these Colodaria, or these radiolarians. And so it's about to happen here, and there you see that the chlorophyll is spiking on the fluorometer, and so this water column is just completely overrun with these things. and it's basically blocking the light, but through about 25 centimeters of water. And so we're really excited to kind of compare this information to between these years and actually try to understand what are these key processes leading to these blooms of different organisms. And so with that, I'll just thank all these people who helped make this work possible, especially NSF for funding our work, and the crew and captain of the RV Savannah. And so with that, I'll take questions.
Adam, anyone have questions for Adam?
Yeah, thanks. Excuse me. Thanks, Adam. That was really innovative. It's great to see those kinds of platforms being developed and making them quicker, cheaper, faster, better. Congratulations. But you alluded to the fact that the shrimp can perhaps see the instrument coming towards them, and they can dodge it. Did we able to? I guess maybe that's where the nighttime toes. Are you able to assess what kind of net or gear avoidance the copepods would show towards this?
Well, the copepods aren't quite as fast as some of the larger shrimp. So our size threshold was 2.2 millimeters equivalent spherical diameter. So it's a little bigger than most copepods. And the shrimp are just really pretty explosive swimmers. And copepods are too, but they're just not quite fast enough to get out of the way. But it's an interesting question. I mean, gear avoidance, I think, is just kind of a fascinating topic in general. And it's really hard to know what you missed and what you got without, almost like equipping something with many different sensors or experimenting with different tow speeds. I think that would be kind of cool to do. We just haven't been able to do it. But with the bigger shadowgraph systems, we tow them typically at 5 knots. And so it's a little faster than our 2 to 3 knots with our smaller system. And even with that fast speed, you see shrimp that are clearly like swimming with the vehicle, and they're all blurred because they're able to really book it.
Do you see any fish eggs in that system? Or larvae?
Not that we definitely see fish larvae. The fish eggs, it's really hard to tell. We do see some egg-like things, and it's just really hard to tell if it's a fish or something else. But yeah. That's a good question.
Anyone else? Adam, are these radiolarians the strontium sulfate skeletal ones, or are these just, I'm asking for Herb, or are these the silicate test ones?
I'm not totally sure. They're clearly, the collodarians are photosynthetic, and I don't know what, like if they leave that shell-like thing on the bottom when they eventually sink out. It's a good question. There's been some recent work on colodaria with imaging systems, and they do appear to be kind of a pretty important organism in a lot of open ocean environments as well. But yeah, I would have to follow up on that with Herb.
Okay, thank you.
I'm not a radiolarian expert.
Right, right. All right, well then, with that, okay, Frederick.
Just a technical question. How small a particle can be detected with your equipment?
So our
the threshold,
our pixel resolution is 44 microns, and so I think realistically you need at least 10 pixels to say anything about an ID. So I say conservatively 500 microns is about the smallest. I would be comfortable saying that's a copepod or something like that.
Okay, well, thank you, Adam.
Thank you.
Yeah, and Dr. Souza is up next. Adam has your.
I've heard this morning about Dr. Baldini saying how little we know about the Brazilian shelf system. Well, the Yucatan Shelf area or Yucatan Current area is soon less studied. Everybody talks, when they talk about the Western current, we talk about probably the loop current and the Gulf current out here. We pretty much never talk about what's happening in the Yucatan Shelf, the Yucatan current. So it's very little work of what's happening here. And why are we interested in Yucatan about this is that we have a region which is completely different to where I ever worked before. This is a white shelf, which I know white shelves, 250 kilometers from traveling north. What's different to me? It's microtidal, and it's oligotrophic, right? And it's not wind-driven upwelling happening here. There's not enough wind to have upwelling. Nevertheless, this area has the second or third largest fisheries of octopus in the world, right? And you have a lot of grouper fisheries happening in here, which is kind of the main local things that are happening together with tourism. Tourism, a very important thing for tourism is in this area where the upwelling is actually happening. We have Holbox Island here, where it's where your basking sharks, your whale sharks go there. So you get a lot of tourists from all over the world around May coming here. And depending what the upwelling is doing, is they will come to Holbox or they'll go back into more to the Caribbean side. So that's kind of the interesting thing, both in kind of what's happening with the economy in the area, but also with the dynamics of what's happening here. So this is how it looks, if you want. This is a picture from the 3rd to the 9th of June using MODIS, looking at SST here and chlorophyll in this area, or quasi-chlorophyll. And then we have the current from HICAM. And you have this strong current coming here, which is up to order of 1.2 meters per second, hitting the shelf area in here, which, if we remember, sorry, everybody said, it's a very shallow area. Everybody is not as shallow as your South Atlantic bight, but it's probably an average about 50 meters. And its slope is very, very reduced. So Alex Kursing could tell you how much you have to travel to get to kind of 20 meters of depth and whatever. It's quite a struggle to get there. But what you see is you typically see this area where you have colder water, cooler, I would say. What's our water temperatures here? They're about 30 degrees, yeah? Where I come that, and this is gonna be about 25, 26 degrees. For me, that's always been warm. Now we call it cool water. But that's where the cool, full of nutrients water comes here, and you start having a bloom coming in this area. I have even a better picture in my phone where you can look at this from last week. And you have this. And one of the things that you have is that this kind of, a little bit of the very coastal area might be polluted with sedum, but you have always this kind of area happening here. So you have all this transport of these nutrients happening here. Other characteristic here is we don't have rivers, right? So all our fresh water comes from underground water, which comes very close to the coast in sinkholes coming out, or blowholes coming out from the water, from the bottom. So we don't know much of what's happening there, but we can detect how it's happening. We're starting to look at it using tracers, because it's continental, it's coming from kind of what's happening down in land. You're starting to look at tracers like caffeine, so you can see where the disposed water is coming out in the area. But it's very coastal. It's very coastal. You have things more out here which are more related to the upwelling. What other problems that we have here? We have harmful algal blooms, and we have two kinds again. We have one that is driven with the upwelling, which is a dinoflagellate, which is poisonous. And then you have another blooms that are diatoms, which are coastal, which are from these nutrients coming from inland. and that's a different thing. That when you get so many phytoplankton that they clog the gills of the fishes and they die. This is a picture of a cruise in September 2011, and what we're looking here is at the depth of the isotherm of 22.5, which is kind of being defined as the Caribbean subtropical underwater, which is the characteristical water that comes up and brings all the nutrients. And you have it kind of in this area very, very clearly developing. But this kind of, then it goes probably not at the surface and it gets transformed because we have very hot weather. We are in the tropics. And then you cannot see it in the surface. But this is reaching the surface. This is about 10 meters in the dark blues, 10, 30 meters deep. So you're getting the water right on the shelf. And this is full of nutrients and all stuff. So that's what we are getting. And then it gets transported. And if you want to know, so this is the picture like everybody has been looking. This is a upside down picture. So this is the north, this way. And we have the Yucatan current coming parallel to the coast. And what you have is a bottom Ekman transport upwelling, which is when it hits the Cabo Catoche, Catoche Cape, it can bring that bottom water that we've just been seeing in other of our areas. And this brings all the nutrients and make this area kind of possible to live for the animals and plants. Pictures of concentrations looking at a different, in the same cruise that I showed of September 2011, we have a lot of ammonia here and a lot of nitrate coming on the shelf from the upwelling area. Yeah, and this is the main nitrate that is coming into the shelf, and then it's kind of get transported and transform. If you look into the shelf area, which is not in this picture, you will see more ammonia in here. And then you have like transitions of what different species you're having. But it tends to be your oceanic species of phytoplankton in this area, and then your oceanic, your more shelf kind of in the other part. This what it's typical happening, and what we think is happening is we have here the bloom here, where we have mainly diatoms, dinoflagellates, and cyanobacteria with phosphate limitations, and then it kind of in the other side of the shelf, you have more dinoflagellates, diatoms, and you have nitrate limitations in that part. And that's what we're looking at studies, and this is work from a master's student that we graduated a couple of years ago. But that's what we're looking at, what's happening, and with tracing kind of the characteristics of the animals and everything, and the water characteristics, we've been seeing that pretty much all this water is coming from this side. Furthermore, if you go to the poster session, you have Alex Kurtzing poster, which he'll explain you more about this. But what you see here is from July 2018. You see your sea surface temperature from satellite images, and you see the cold water all along the coast on the shelf, the upwelling area. This is the sea surface temperature anomaly, so you take the mean temperature, and this is your sea surface temperature. So it's not only kind of having the shelf part of it, but you also, or the northern shelf part of it, of the Yucatan shelf, but also in this side, which is the Campeche side, you also have this water that's coming from far away, having their characteristics. And there's no way that you can get these temperatures from other area that's here, because all the rest of the Gulf is warm. It's a lot warmer. And again, this is pretty much stolen from Alex's poster, and you can see this is Campeche, which will be like this part of the coast. And these are surveys that have been done measuring over the area. And you can see how we have this relatively colder water getting into the To the coast, this is surface temperature. So you see 30 degrees temperature, you have 28, 29 degrees water coming here, which will be characteristic of modified upwelling water. But if you look at the bottom areas, if you go to 7 Kui, which is this area, you start seeing in the bottom, you have very, very cool water. I won't say what very cold because it's just like 25 degrees, but you're still having these 25 degrees water present into the system all the way around. This is going to attract you a lot into what this area, but forget about it. The important part is against this shelf. Its salinity is it says 1 kilometer, I just checked. It's a 2 kilometer global MIT model from September 11 to November 12. It's sea surface salinity because it's what I managed to get from the bacterium. But you can see all the transport coming this way here. And not only that, you can see all the microstructure happening. Somebody said turbulence and microstructure are important. They are. And I learned it from my very beginnings, but probably for that I need the mouse, don't I? Just click. Okay. So you see how it's coming this way, and it gets all the way down. Either, yeah, all the transport, and all these little scale happening there, which I can't remember who I was talking with. We were talking about how the variability is important in the transport. Okay, I'm going quicker. So you can see that's how it's get transported from one side to the other. Oh, I missed one slide. Well, never mind. So I'll leave it with that then. But basically, that's what we are where we are looking at how these. There's several slides missing, actually. I'm worried now. Oh, shit. Which it was looking at the variability itself. Well, I'll have to wing it now. What you see is like, if you look at model data output, and I can show you on my computer, because I have the full presentation. If you look at the currents, your mean current from NEMO model Orca 12 at a degree, you have the mean transport coming this way, pretty much looking at the loop current and the Yucatan current. And if you do the EOF of that, the spatial EOF, the first mode is that way as well. You have that shape and then slightly transport. And you get the variability driven of how this current, as we were told before, as it gets closer or further away, you have either upwelling or downwelling of the shelf area, and that's what it's driving it. At the same time, that has an annual variability, which you also have in the temporal EOFs of temperature. You have this very annual variability, which well, when I first did it in the temperature, and I thought it was the seasonal cycle. But looking in the currents, I think differently now. The second mode of temperature, although very small, you have very interesting variabilities, which appear to be related to more things like the northern winds that we have present, and that happen, the northern storms that happen in this area. I'm really sad that it didn't look at, but if you are really interested, come and see me and I show you the EOF process from there. Another thing, just finishing now, because I have less than a minute, if you want to see like the variability of the entire area related to NAO and ENSO, there is a poster back at the end where you can have a look, and then we also can talk about the variability here related to NAO and ENSO. Thank you.
Anyone have some questions for?
What's the variable you show in the movie? Is that
That's sea surface salinity.
sea surface salinity?
Yeah,
Simulated.
salinity.
But how about the satellite observed salinity?
The satellite observed salinity won't be that good in that part because that's usually for further off the shelf. The salinity from satellite is not very good in shelf areas.
Okay. Another question is that could you put on the picture of the SST and the chlorophyll?
No.
I see that the SST, the cold SST, is only concentrated on the northeastern Yucatan shelf, not the southeastern Yucatan shelf. Over the entire along the coast, the chlorophyll concentration is high, but the SST actually is also high. Yeah. So see, the cold temperature only concentrated in the very small area. In the large area, the chlorophyll concentration, could it be from the river runoff that input the nutrient or stuff
There's no
like
river run.
Yeah.
There's no river.
There's no river.
There's
Okay.
no river.
Then how
Everything is too close. This is it's upwell. It's very localized if you want, but it comes to this if you want to have a look. That in that image is very localized. This is a different time, but you see all these areas
along the entire coast. The chlorophyll.
This is you can have it entire coast. I'll show you the EFs and the data if you want, but it gets over here. But you have to remember that from the time that it comes from here to here, you have enough time for it to radiate it and warm up. Yeah, it's very high temperatures that we have in this area, so you have a lot of heat transfer. But you have these very localized areas. Well, very localized. This is about 50 kilometers wide. So, well, it's even more. This is 200 kilometers. But if you say that that's about 100, if you say it's 50 kilometers, that's 3 spacetrimps of transport that you're having this way according to bottom Eggman transport. So you're going to have a lot of water coming into the shelf. And then it gets transported. If you look at long time series from well, at least model data, you will see a band of fresh water, cold water along this area, which is permanent or
So it takes 1 to 2 weeks to convert those nutrients into phytoplankton. So there's a trophic delay as you go along the peninsula from east to west. So I think there's a time factor in that. The nutrients are powerful.
So in the South Atlantic Bight, we see that the nutrients peter out at about 20 degrees centigrade, but you're seeing nutrients in the water at source waters of 23, 24, 25, right?
Yes.
What depth is that actually originating at?
That's probably coming from about, 200-300 meters. Okay. Yeah, 200-300 meters.
So that's equivalent.
Yes.
It's just warmer. Yeah.
Yes. Because it's subtropical Caribbean underwater. It's getting a bit warmer, and it gets its speed pretty much to the same. And I say, yeah, for us that's cold because it's just so hot in there. It's like you get 30 degrees water temperature. It's a bathtub in there.
Right.
So.
Okay. Well, let's move on to our next speaker, who's Dr. Yuan.
Oh, yeah.
Phenomenon called CPF, and I also talk about the variability and the global warming. The ocean circulation in the Yellow Sea, East China Sea in the past has been mostly based on conjecture, and the information is not very accurate. For example, the circulation along the east coast of the China, we call the Yellow Sea coastal current. Actually, it's not a coastal current. It's not following the coast. Different people also call that the Yellow Sea coastal current. I'll show you that that's not really accurate. And so the question we want to understand is how does the crucial exchange with the shelf, particularly the inner shelf of the Yellow East China Sea, and what's the dynamic and how large are the transport and the carbon effect? When I was at NASA Center, I noticed that this in the Yellow East China Sea, each winter, from fall to winter, there's a large area of high sediment, suspended sediment concentration. You know, the Yellow Sea, why is it called yellow? Because of concentration of the suspended sediment from the Yellow River. And you can see that from fall through winter, the entire Yellow Sea is yellow, okay? But in the summer, when the Yangtze River runoff is the highest, actually the suspended sediment is very slow. And when I was at NASA, NASA called this yellow plume, the Yangtze River plume. Actually, it's not right because it's from the old river, Yellow River mouth, from the Subi coast, which is the area that I show you, the Yellow Sea coastal current. Now, the concurrent measurement in the offshore of the Yangtze River clearly shows that there's a salinity and temperature front offshore of the Yangtze River, which prevented the nearshore suspended sediment from going offshore. And the current clearly along the coast is to the south, but offshore in the canyon is to the north. And the image clearly shows that you have this plume that's going offshore. The winter salinity climatology, we don't have satellite image at that time. And you can see the temperature image also shows this offshore movement from the Subi coast to the continental shelf southwest of Jeju Island. And so we name this current East China Sea current because this name is not used in the area. And you can see that that was clearly marked by the high suspended sediment moving offshore, and part of them returned to the Yellow Sea trough because of the intrusion of the Yellow Sea warm current. And then, how do I return back to the first? You see the dot in the Okinawa. Okay, you fly one. Yeah, you see there's a sediment trap in the Okinawa trough here that has the record shows that the suspended sediment, the capture is highest concentration in the middle and the bottom part, but not in the surface, suggesting that this sediment, measured sediment, is not from the atmosphere aerosol. It must be from the ocean. The transport is subducted into the Okinawa trough. And it is highest in winter, from fall through winter, and the lowest in summer. And we see the satellite trajectory on the cyclonic side of the Yellow Sea warm current clearly goes offshore in the southeast direction. And in that winter, I have cost money to launch this cruise in winter, which is rarely in this area because of the monsoon and the weather is really rough, the sea is rough. And we see the measurement of the ADCP current clearly shows that the current is offshore. The current here, this point, the current is offshore, and all the surface drifter trajectories launch the jog at 15-meter water jog moves offshore, countering the Ackermann transport of the surface wind. The dynamics, I could spend the entire talk talking about the dynamics, but simply when the northerly wind, the monsoon build up, the sea level build up to the China coast because of blocking of Ackermann transport, then this propagation of continental shelf waves quickly set up a southward pressure gradient across the shelf that basically guided the offshore transport from Subi coast to the crucial edge. So this is the East China Sea current, or the cross-shelf circulation. Another phenomenon very important is called the cross-shelf penetrating front. This is also observed off the Mid-Atlantic Bight coast. And you can see that the chlorophyll concentration in October 2000 offshore of the China Yangtze River, south of Yangtze River, actually Chinese Minzhou coast, shows a fish head shape of the chlorofluor concentration that extending offshore in the southeast direction by a large distance over like 50 kilometers to 100 kilometers. And the SST image shows that that's nearly in summer, but it's fall. In October, the weather is still very hot. So that SST shows cool signature. And then I was lucky enough that I was able to direct the ship when we observed a front, although it's not very typical. It's a small front. So we sent the ship to the area and then we measure it. We see that it is a freshwater plume, and there's a contour current below the surface offshore excursion. And so we confirmed that this is produced by current. And that's the example of the CPF. You can see that it's frequently occur off the Chinese coastal Minzhou coast, and it constitutes a significant offshore transport. Its mechanism is not unknown. Is not known yet. So the seasonal cycle shows that it's smallest in spring, but the highest in summer and winter, summer through winter. And the interannual variability doesn't seem to be very phase-locked with ENSO. So we hypothesize that there are two pathways. One is the cross-self-transport, the other is the ensemble transport of the CPFs. In winter, the aspignal shows that the crucial water is warm over the continental slope, so that the shelf water will go beneath the crucial thermocline. That forms a mechanism for carbon sequestration, which I'll talk about in the next slide. So we use a global model with high resolution in the area. Actually, this model is collaborated with the University of Hamburg and Max Planck Meteorology Institute. Their model was able to resolve the shelf. You can see the grade is high enough. And we were able to simulate the current over the shelf with reasonable accuracy because we compare with historical measurement. Based on that, we calculated the cross-shelf transport across 100 meter and 200 meter isobaths, and we also estimate the carbon transport. You can see that across 200 meter isobaths is not a good criteria because the transport was determined by the difference between the Tsushima transport and the Taiwan Strait. So we use 100 meter because it's completely from the closed box of the shelf. And the transport of the carbon is about 110 and 0.1 million tons of DIC, DOC, and POC across the 100 meter isobaths to the crucial in winter, presumably to the subthermocline of the crucial. And based on the global estimate of upwelling, that would take roughly about 100 years to come back to the surface. So that would form an effective carbon sequestration and burial. Now this, we also calculate, because they have projections, different scenario. We have estimated like 2 or 3 scenarios, I think 2. So we see that all this offshore transport and carbon, some of the carbon, I think, offshore transport, they are increasing, and different global warming scenario, and the increasing rate is highest in winter. And with the concentration, the measured concentration, which we know so far, we don't know how the carbon concentration will vary in the future, but we assume that the carbon concentration will fix. Only the transport will change. And then we can estimate the cross-shelf transport, which shows they all increase in the future in the next 100 years or so. The paper is already published, and the details, I think that this limited time I cannot cover. But I want to emphasize that the reason we use a model to calculate the cross-shelf transport is to calculate the air-sea carbon dioxide flux. Because the advection, you have a C bar multiplied by the convergence, which is 0 of the volume, and by the C prime multiply the anomalous transport. If in the measurement, you use ADCP measurement, if the volume is not conserved, then your estimated carbon dioxide air sea flux would be erroneous. And you can see my colleagues estimate the transport is significantly different from us. And I think this is a new estimate, but in the past, in the China East China Sea, Yellow Sea, they said that this is a weak sink of carbon dioxide. Our estimate, although we only based on 4 cruises of the carbon measurement, our estimate is very high, and same order of magnitude as the Southern Ocean. The Southern Ocean, I think, is like 50. The number, I forgot what unit. I never always know, because I'm not a chemist. So the number is 50. Ours the number is 13, something like that. So it's pretty high. That's our philosophy. And last but not least, I want to mention that you can see that the Mid-Atlantic bight circulation and the Yellow Sea, China Sea, they are very similar. They have the cross-shelf transport, cross-shelf front, the cross-shelf penetrating front. They also have the intrusion of the Gulf Stream, and some of them may be subject to the same dynamics as the arrested topography wave and also some other interactions. So I think some kind of a comparative study or collective study or cooperative study can be organized if the group of people should be interested in. What's going on? Okay, thank you. And that's the conclusion. Basically, we found that major cross-shelf current and cross-shelf penetrating front exists in the Yellow East China Sea fall through winter. And we estimate the carbon transport. We think that this transport is going to increase under global warming. This cross-shelf transport significance suggests large air-sea flux, which has been overlooked in the past. I think that there are unresolved issues, which are the CPF dynamics. We have some hypotheses, and we have some preliminary results, which I cannot cover in this talk. Yeah, I think the global scale cross-shelf transport so far has not been estimated. And if this audience or the group of the scientists should be interested in collaborating with us in estimating the global cross-shelf transport of carbon, please talk to me so that I have some idea using the model and some measurement to do a collaborative work, cooperative work, which I think is going to be a significant publication in a high-impact journal. Thank you.
Are there any questions for Dr. Yuan? I'll point out that last image that you showed of Lee and the cross, that plume is from North Florida, so it's really the South Atlantic bight, not the Mid-Atlantic bight.
I'm sorry, South Atlantic bight.
Yeah.
Sorry.
I just want to give us credit where credit is due.
Yes, yes.
So that the offshore transport of carbon down theoretically cascading over the shelf edges, are you talking entirely about inorganic carbon, or are you talking about particulate carbon being
I have calculated all of them, the DIC, DOC, and POC. Three of them. DIC is the largest, and particularly the volume conservation is the most significant, because if you should have unbalanced volume, then you have multiplied by the mean DIC concentration, that's going to generate significant error of the carbon transportation.
So this is something that we've discussed here without having it at Skidaway. So as that water moves over the shelf and seeks to find its own density, does it maintain itself as a coherent water mass, or is it mixed strongly with the Kuroshio as it passes by?
That's a good question, but I think it's an identifiable water mass below the Kuroshio thermocline, because it's fresh water from the shelf, and that's easily identified. We just do not have the measurement in the Kuroshio area, and the Japanese now do not allow us to measure. Yeah.
Yes, because certainly, if it maintains its coherence as a water mass and it dips down there to 300 meters,
fresh water.
It's much easier to imagine it sequestering carbon for a more extended period, as opposed to losing its identity and as it mixes with the Kuroshio passing by.
Yeah, some mixing will happen, but it will stay in the subclass because there's a lack of mechanism to come up across through the subclass. Yeah, it is. I think it should be an identifiable water mass because of the freshwater characteristics.
All right, thank you. Now we are. Oh, Lisa, what's? Sure, there's always time for another question.
Thank you for your talk. I have a question. So on your conclusion slide, my interest was very piqued by, you say that your expectation is that the cross-shelf transport will increase over time. And excuse me if I missed you explaining that, but can you quickly explain why that's your expectation?
That increase is mostly because of the increase of the cross-shelf transport under the global warming forcing, because of the global warming has produced the wind force. I think we also have the heat flux stuff and precipitation river runoff in there. So everything's there, but it was forced by the global model, coupled model, the condition, and then it produces larger cross-shelf transport, and then multiplied by the the current concentration, it will generate higher transport. But you can argue that maybe the concentration will get lower so that it will compensate for the large cross-shelf transport. But we don't know how the carbon concentration is going to change in the future. We do not have that information. In that case, you have to run a full coupled model, including the carbon cycle, to estimate that.
Okay, we have a 20-minute break scheduled. Let everyone. I heard the clinking of glassware in the back, so there are some coffee and refreshments. And thank you. Okay, our next talk is going to be by Dr. Asufo Halo from Cape Town, South Africa. It's going to be a recorded talk. We have no idea if he is actually going to show up for Q&A since it is what? It's 4:40 a.m. in Cape Town right now. He said he would do it, but we haven't seen any evidence of him. But anyway, Wayne, you want to get that started? Influence of the Madagascar Ridge on circulation in the Agulhas current system.
Good evening. Thank you very much indeed for the privilege that you have given me to present this theme in this important meeting. My name is Isufu Halo, and I work for the Oceans and Coast Research Group in the Department of Forestry, Fisheries and the Environment in Cape Town, South Africa. On behalf of my co-authors, I will be talking on the topic, the influence of Madagascar Ridge on the variability of the flow in the Agulhas current system. This slide shows the bottom topography in the Southwest Indian Ocean. This is in the shading. It ranges from very deep ocean basins, represented by blue color, to a very shallow areas, represented in red. And the overlay, the arrows are indicating the main oceanographic features in the area. The circulation is dominated by a number of mesoscale and large-scale structures, mean currents, and the eddies. Through the Mozambique Channel, there's a number of mesoscale eddies in form of cyclone and anticyclonic eddies that migrates to the south. South of Madagascar, Southeast Madagascar current, upon passing the southern tip of Madagascar, it generates vortex structures, dipole structures that propagates to the south. When those structures that come from the Mozambique Channel combined with those coming from the south of Madagascar flows into the Agulhas current region, they do influence quite significantly the variability of the Agulhas current. The Agulhas current retroflects. It rotates on itself on about 40 degrees south and 17 east. During that process, the bulk of waters goes back into the Indian Ocean, there's Agulhas return current, and parts of the flow go into the South Atlantic in form of mesoscale structures like Agulhas rings. My particular interest with this presentation is to highlight the role that the Madagascar Ridge plays on the variability of the flow. The Madagascar Ridge is represented here by this red box. It's just immediate south of Madagascar. It lies on the north-south orientation and covers an area that spans from 42 to 47 east and the 26 to 36 south, and gives approximately to 600,000 kilometers in terms of width, and the length is 1,300 kilometers. The region raises to about 3,000 meters from the seafloor, so it is quite shallow. So it prevents the mixing between waters from the eastern side, which is the Mascarene or Madagascar Basin, from mixing from waters from the Mozambique Basin, which are located in the western side of the ridge. So the flanks of the ridge, the eastern and western, they descend quite steeply towards the seafloor. So previous studies in the region have implicated the presence of Madagascar ridge as being controlling the characteristics of the local hydrodynamic conditions. When we come across with studies such as those published by Matano in 1999, Cotley in 2006, Zidler in 2009, even our own study in 2014, all these studies, they converge, they support the idea that the Madagascar ridge is an important bathymetric feature that modulates the flow in the area. In fact, this finding seems to be corroborated by analysis that we made by identifying mesoscale structures in the area and the mapping upon the seafloor topography, as is illustrated in this map that I show here to my left. So we have computed 2 years of satellite data, and so just for visualization purpose, we have shown these 2 years, but of course we have covered the whole altimetric period. We identified the cyclonic eddies, which are represented by these blue dots as the eddy generation sites for cyclones. And the bold red dots are representing places where the anti-clockwise rotating features are generated. When we do overlay these with the topography of the Mascarene, of the Madagascar ridge, we see that much of these eddy generation sites, they sit, they're quite well aligned to the places where the gradient of bathymetry is quite intense, really suggesting that the influence of Madagascar ridge might impact the mesoscale variability in the region. But now, a couple of questions have been put forward. The first, how do the Madagascar ridge impact the volume transports across the main ocean currents in the areas, in the Agalas system? The second question is, how do Madagascar ridge controls the eddy characteristics? Is Madagascar ridge a platform that enhances eddy production, or it influences the dissipation of the eddies? Well, to find the answers to these questions, we have used a three-dimensional ocean model. This is a CROCO model, which is an evolution of the well-known regional ocean modeling system that the French researchers have developed. We have run this model using climatological monthly means, both at the sea surface and at the lateral boundary conditions. The sea surface comes from COADS, which is relatively low resolution data. And at the lateral boundaries, we have used also an Australian database that is a CARS 2009. 2 simulations were performed. One that we call the reference experiment. We have run the model within general bathymetry of the ocean, the GEMCO 2014. The second experiment that we call idealized experiment, we have removed the Madagascar ridge by flattening it towards the seabed. And the interpolated bathymetry are shown in these maps that I show. On the top, it is the reference experiment, and in the middle is the idealized experiment. If you look carefully to the region south of Madagascar, you will see that the Madagascar ridge has been removed. Of course, to make sure that these changes did not affect the topography elsewhere, we have made some sensitivity studies. And after finding the right tune for that, when we made the difference between our reference experiments, the topography on the reference experiment, with the bathymetry in which we have modified, we see that the change, it is only around the Madagascar ridge. So this gave us the confidence that we could run that topography to simulate the flow field in the absence of Madagascar ridge. And this is what we get, the results. So we have shown here just the root mean square of the sea surface height, which just gives an idea about the variability of the flow. We have computed this from the 2 experiments, and we compare against the variability that we get from the satellite observations. The satellite observation is represented on top. In the middle is the reference experiment, and below, in the bottom, is the idealized one. Couple of consistent patterns that we see, both the Mozambique Channel and south of Madagascar are indeed sources of the variability towards the Agulhas current. So the simulations are able to reproduce this, and the simulations also are able to reproduce the right location of the Agulhas retroflexion and the eastward movement of the Agulhas return current along 40 degrees south. However, when we look carefully to these diagrams, some differences are also quite obvious. We see that the both simulations have overestimated the variability compared to the satellite, especially on the Agulhas current itself. However, we bear in mind that the satellite data is relatively less in terms of the resolution. The graded resolution is weaker than the simulated products. Nevertheless, when we do compare the simulations among themselves, the reference and the idealized experiment, we see that the reference experiment produces higher levels of variability on the Agulhas current and slightly less on the idealized experiment. But overall, quite consistent patterns are present. Now, when we look at some results by comparing the idealized and the reference experiments, we computed the volume transports just to see how the Madagascar ridge affects the transport across the main ocean currents in the region. We have computed the depth integrated transport from 2,000 meters up to the sea surface. The reference experiment is shown here on my left on top across the whole simulated domain. And the reference experiment is the middle panel. When we look at the differences between these two, with the naked eyes are not quite obvious. However, the only difference that can be easily spotted is the region south of Madagascar. The orientation and the structure of the streamline of the volume transport has changed. Overall, it suggests that in the bigger picture, the large scale doesn't change much, but localized features south of Madagascar did change. Now, to corroborate this, to quantify these changes, we have made the differences between the reference experiment against the idealized one. And the result of this difference is presented on the bottom here. Looking careful to this image, at the bottom, we see the difference, 10 severe groups, region south of Madagascar. We see also on the recirculation of the Agulhas current, 10 severe groups. On the Agulhas retroflexion area, we see increased 10 severe group, and the same is true along the path of the Agulhas return current. So that increase of transport that comes from the south of Madagascar right into the Agulhas current doesn't reflect much on the amount of leakage, amount of water that goes into the South Atlantic. Rather, it results on that recirculation into the Indian Ocean via the Agulhas return currents. Then we have also looked at the time series of the volume transport just across the width of these main currents. The panels on my right, the top one, is a transit that was made across the width of the southeast Madagascar current at 23 degrees south. The second from the top, it is a cross-section of the transits that we have computed the transport across the width of the southeast Madagascar current when it runs zonally across the Madagascar ridge. It is around 45 east. And the third from the top is the transport across the Agulhas current at 32 south. And finally, the one on the bottom is the transport that we have computed across the path of the Agulhas rings when they migrate into the South Atlantic. Looking at the patterns of these transports, major differences that we see between the experiments, between the reference experiment and the idealized experiment can be seen on the power spectrum. This can be seen in the area south of Madagascar, exactly where we removed the ridge. In these diagrams, so the blue is representing the idealized experiment, the black is the reference experiment. So we see that the significant variations or differences between the two experiments is in the region south of Madagascar. There's a decrease in terms of energy levels. And much of these differences takes place on the processes on a frequency of about 1.5 to 4 per year, which suggests that much of these processes that are being impacted by removing the Madagascar ridge are processed across the mesoscale range. To consolidate this, we have run an eddy tracking algorithm in the two experiments, and we made an assessment. Just for visualization purposes, we just represented these 2 years of data to facilitate our inspection by eye. The reference experiment is on top and the idealized on the bottom. Looking at the area south of Madagascar, it's quite clear that the number of eddies have decreased in the idealized experiment. Again, the red is representing the anticyclonic eddy generation sites, and the blue, the cyclone, clockwise rotating eddy generation sites. To validate further this, we have computed the eddy presence, the frequency of distribution across the whole simulated domain. To the left are representing the outputs from the reference experiment, and to my right are the idealized experiment. The top panels present the cyclonic structures, and the bottom panel shows the anticyclonic structures. Looking at these, we see that the reference experiment in the region south of Madagascar appears to produce more anticyclones than the idealized experiment, these two maps on the bottom. When we compare the cyclonic distribution, it appears that the cyclonic eddies are almost everywhere in the idealized experiment. We selected these areas. One to the south of Madagascar, where bulk of these changes have been observed, and the one exactly on the Agala's retroflexion region, where our results suggested that no significant changes have been observed. We looked at the eddy density distribution in those boxes. The top one represents the boxes south of Madagascar on the ridge domain, and the plots at the bottom represents the box in the Agala's retroflexion. Again, the reference experiments are on my left and the idealized on my right. When we compare the region south of Madagascar, we see that on the reference experiment, the frequency of eddies occurrence did peak on eddies with a size of 50 kilometers radius for both cyclonic and the anticyclonic. While in the reference experiment, the peak was higher for both cyclonic and anticyclonic on eddies with a radius of 60 kilometers. It shows a slight increase in size by removing the eddies. South of the Agala's current on the Agala's retroflexion area, no significant change in this regard has been observed. Both the peak of cyclones and the anticyclone appears to be centered at the scale of 40 kilometers in radius. Now that we have defined that indeed the bulk of change are centered around south of Madagascar, where we removed the topography, we have investigated the eddy properties in the region south of Madagascar. And the result of our analysis are shown in these histograms. We see that the high eddies with the highest amplitude on the reference experiment, the cyclones centered at the 50 kilometers and the anticyclones at the 60 kilometers. When we look at the non-ridge experiment, the idealized, both cyclones and anticyclones had this higher production or higher amplitude for eddies of 60 kilometers. When we look at the relative vorticity between these two simulations, we see that the reference experiment shows higher levels of vorticity, higher intensity in cyclones and the anticyclones at 50 kilometers, while in the idealized experiment, the peak on the relative vorticity is at 50 kilometers for cyclones and at 60 kilometers for anticyclones. Now, when we compare the reference against idealized experiment, we see there's a slowdown, there's a decrease on the intensity of vorticity, clearly suggesting that when we do remove the Madagascar ridge, there is a spin down of the relative vorticity. In conclusion, what can we say? Well, these results are preliminary, but the following points can be put forward. One, the presence of Madagascar ridge decreases the volume transport across the Agalas current, Southeast Madagascar current, and the Agalas return current. There's no obvious change on the amount of the Agalas leakage. The second take-home message is that the presence of Madagascar ridge enhances the variability of the flow across the mesoscale range, results on an increased production of mesoscale eddies in the region south of Madagascar. Overall, the highest peak of the eddy density distribution revealed smaller eddies with the higher amplitude and stronger relative vorticity in the reference experiment than in the idealized one. This suggests that the Madagascar ridge influences the geometrical and the dynamic structures of these eddies. My recommendation, the recommendation that I put forward is perhaps to conduct this study on a much higher spatial resolution model to see if smaller scale processes will be able to actually bring new results. Thank you very much for your attention.
Okay, so he decided to stay in bed, which is entirely reasonable. So we'll move on to Robert Todd then.
Robert, who would prefer to be in bed by about this hour, so this will be interesting. So I'm going to talk to you about shelf deep ocean exchange near Cape Hatteras from 2 years of underwater glider observations. This was part of the Peach project. That was an NSF-funded project, fieldwork from 2017 to 2019. And I'll tell you a bit more about it in a second. So what Peach stood for, Processes Driving Exchange at Cape Hatteras. I will take full credit for the tortured acronym, but it was created while here in Savannah, out at Skidaway while we were planning the project. So Peach was an appropriate thing here in Georgia. Overarching question for Peach was, what are the dynamics controlling exchange between the shelf and the deep ocean, and focusing in particular on the Cape Hatteras region. I'll tell you a bit more about that in a moment. The field work for Peach was intentionally multi-platform and multi-scale. It spanned 18 to 24 months, depending on which platforms. And you can see on the map there the variety of things we had. We had 2 types of gliders in the water. I'll talk about just the spray gliders today. There were sea pies on the sea floor below the Gulf Stream and along the edge of along the continental slope. There were moored ACPs and CTDs along the shelf break. Quote-unquote tall moorings on the shelf. They were 30 meters tall because the water is only 30 meters deep. And then we had 2 kinds of HF radar systems along the coast, giving surface currents over this hatched region. And of course, the ship was down there 3 times doing turnarounds for all of these moorings. And then there was also a modeling component run primarily at North Carolina State. Had both idealized and realistic nested assimilating models. And so all of these institutions here were part of it, and a bunch of us PIs are here in the audience. So this quick outline, I'll just breeze on to the overview of the Peach glider observations. So for those that are falling asleep, just stare at the squiggly lines, it'll repeat. That's what the spray gliders I had in the water did over a period of 2 years. So the gliders, for those who aren't familiar, they're buoyancy-driven platforms. They're not that different than taking an Argo float and giving it the ability to change its orientation and give it wings. So as it goes up and down, it's also able to move through the water column relatively slowly, about 1/4 meter per second horizontally. We did 7 back-to-back deployments for Peach. I also have, so those are the gliders doing the tight L-shaped pattern along the edge of the continental shelf and across the outer continental shelf. And then you'll see these other gliders swooping in from the south that are zigzagging across the Gulf Stream as part of another project. And all of the gliders had CTDs, ADCPs, chlorophyll fluorometers, and some of them also had dissolved oxygen sensors. So what did we accomplish? So for the purpose of this analysis, I'll divide the data into 2 distinct transects. One is the slope transect, shown in red. Went about 115 kilometers from it, the southern end of it was determined by where the Gulf Stream was at any particular time. The northern end was near 137 north, just south of Norfolk Canyon. And then once the gliders got to the northern end, they took a short little jaunt into the 50-meter isobath across the shelf break. So we'll call that the shelf transect. We had 107 occupations of the slope transect in 2 years, so it's about an 8-day repeat cycle. The shelf transect, we had 73 occupations. Those tend to be pairs of transects, inbound and outbound, separated by about 2 weeks, because that's how long it took the glider to go south and then back north along the slope transect. So we had profiles to 500 meters or deeper sometimes, or to near the bottom of temperature, salinity, velocity, chlorophyll, acoustic backscatter, and some oxygen. And I'm not going to talk about those last 3 here because if you remember, I'm a physical oceanographer and it's dangerous for me to talk about those things, but the data exists. On the right here is just one example transect along the slope transect, and this will orient you to some other figures coming up. So the Gulf Stream, southern end of the slope transect is here, warm salty water in the Gulf Stream. As you move north on the slope transect, you see all kinds of things going on in this particular one. I'll point out this is mid-Atlantic Bight cold pool water. This is water that was up on the shelf, cold subsurface water that's now been exported out into the open ocean. We'll see more of that later. And then we also have this little cross-shelf transect, so obviously deep water on the right, 50-meter isobath. There we go. 50-meter isobath on the left-hand side, and the little tick marks at the top give you an idea of how densely spaced the individual profiles are. So this is temperature at the top, salinity in the middle, and then cross-shore velocity and along-shore velocity from the onboard ADCP. So what do we get to learn about the hydrography and the circulation? So again, another movie to keep everybody awake. That is observations only. That is not from a model. So we have a whole lot of data here, and with some fancy least squares and objective mapping work, we can smooth it out and create a gridded product in 3 of the 4 dimensions. So we have depth, we have a long track, and we have time. We don't have that extra 4th dimension that a numerical model would give us. But this is now running through 2 years of those same variables and now smoothed out. So there's roughly a month temporal filter and something like a 30-kilometer long slope filter applied. So that's all fun to watch, but keep in mind that I have a mean and an annual cycle that I can work with, and then I can compute anomalies relative to that. That will come up later. So about the mean circulation hydrography, so same format of the transects and variables, but now this is the time average over the 2-year period. So what do we see? So first of all, we have the Gulf Stream. It's warm, it's salty, and it's quite quick going to the northeast. So Cape Hatteras, of course, is where the Gulf Stream is leaving the continental margin. We see net export. Whoopsie. Where did it go? There we go. Net export. So this is the cross shore velocity. We see that flow is offshore, and somehow I managed to skip a slide, I think, that was telling you more about Peach. What you should know about this region at Cape Hatteras is that the flow on the shelf is from the mid-Atlantic Bight shelf flow is to the south. The South Atlantic Bight flow is to the north. You have a narrowing shelf. If you have waters coming together, there must be net export. There it is. We see it. That's reassuring. We also see that along-shore convergence. So this is a transect just offshore of the shelf break front, just offshore of the continental shelf break. We see southward flow meeting northward flow at about 36 north. And you'll see that line of convergence come back up in some plots later, just as a reference point. In the cross-shelf direction, we see the shelf break front. We have cooler, fresher water on the continental shelf, warmer, saltier slope waters offshore. But it's worth looking closely at the gray lines. Those are isopycnals. And in the mean, the isopycnals are very nearly flat at this latitude. That's not the case further north in the mid-Atlantic bight, where the shelf break front, on average, is also a density front. Down here at Hatteras, things have adjusted, particularly on the shelf, to the point that the density is very similar on both sides, on average, not necessarily at any given time. Because there is little cross-shore density gradient, the shelf break jet is pretty weakly sheared. Geostrophy tells us it should be so, but there is a southward jet along the shelf break, as we expect. Next slide. Okay, so shelf water export. So these are results from a paper I published a few years ago now. So what I did is I took all of the observations, both the map and the individual transects, and I looked for I defined shelf water to be, this is mid-Atlantic bight shelf water. It's easy to classify because it's low salinity. So anything less than 34.5 in salinity, I called that mid-Atlantic bight shelf water. I'm looking at the slope transect, and I'm counting up the volume of water coming off the continental shelf that's that salinity or lower. So what you see is now a time series of that. The dots and other symbols are from individual glider transects, and then the curve, the smoother curve, is from the mapped product. So the average is about 0.3 sphere drips. So that is actually right what we thought it should be. If you ask someone like Glenn Gorkowitz what the alongshore flow towards the south is, he would, particularly in the shelf break front, he would tell you it's about 1/4 sphere drip, that we get that kind of number for the mean transport. But the standard deviation is also 0.3. So there's a lot of variability. And actually it turns out that over that 2-year time period, half of the total offshore transport happened during 17% of the time, 1/6 of the time. That's telling us this is a very episodic export. It's not steady offshore flow of shelf waters. It's happening in these discrete events. And you can see some of them have exports up to 10-fold bigger than the average. So this particular event here with the star. It's worth noting most of the export is happening during the summer. Sometimes that export of shelf waters is associated with passing tropical systems. Those are the named storms at the top. Sometimes it's not. A little bit of dynamics. So we also have, that was looking at the longshore, or the slope transect. If we look at transport through the shelf transects, and remember this is not all the way across the shelf, it's only seaward of the 50-meter isobath. But that's now transport of waters fresher, the lower panel in blue, is transport of waters fresher than 34.5 through that transect. And without belaboring it too much, there is, first of all, it's worth noting there's reversal of the flow. There's northward flow of those waters 20% of the time. So things are getting all kinds of interesting down there, and the mean flow is going the opposite direction we expect. But there is a reasonable correlation between this signal and the export. So when there's more flow to the south through the shelf transect, there's more export of waters through the slope transect. That makes sense if you think in a continuity sort of sense. And there's also a slightly weaker correlation with the cross-shelf density gradient. So when there's a stronger cross-shelf density gradient, that's in geostrophic terms, that's going to force more water towards the south. All right, a few examples, because we said this was episodic. So what do these episodes of export look like? So here's one example from September 2017. For those in the eastern part of the US will remember there were storm after storm after storm going on the East Coast. Hurricanes Jose and Maria passed close by Hatteras within about 2 weeks of each other. They produced winds down the shelf, so these were storms offshore. The winds so these are cyclones, so then the winds were towards the south over the mid-Atlantic bight. You can see in this series of SST images, you see the cold water of the mid-Atlantic bight shelf being accelerated southward, running into the Gulf Stream, making a hard left turn offshore, and then going out along the edge of the Gulf Stream, which is a pretty typical surface pathway for export. What did the gliders see? So this is one transect through, roughly coincident with this snapshot here. And we saw, if you look particularly at salinity, we see a whole lot of low salinity water. That's mid-Atlantic bight shelf water that's now been exported offshore, and it is moving offshore. Let's see. This is just the wind field. So this was a near surface export event, driven by the wind, pretty clearly. So here's a contrasting example. Whoops. So this was that biggest export event we saw in June of 2018. So it preceded any big storms coming through. It was in excess of 3 spherodrips. That if we had 3 spherodrips of flow going off the Mid-Atlantic Bight Shelf all the time, there would be no water left on the shelf in pretty quick time. So what did that export event look like? Well, so now if we look at temperature and salinity, where is all the shelf water? It's subsurface. In fact, it's that cold pool water, that subsurface water from the shelf, and it is denser than the Gulf Stream water, this warm, salty water on top. So that water has been going out underneath, subducted under the edge of the Gulf Stream. And the reason there's so much transport, look how fast that water is going. Look between the 2 black lines that are outlining salinity. Those are currents, upwards of 50 to 75 centimeters per second. So a subsurface event, it was cold pool waters, and here's the kicker. You're not going to see a sign of that at all by satellite-based remote sensing. So we can see the previous event by satellites. This one was completely invisible satellite. And it was the biggest event we saw in a 2-year period. Going to the other extreme, the winter of 2017-2018, there was almost no export for 3 months south of 37 north. So the southern 115 kilometers or so of the Mid-Atlantic Bight, there was no water going offshore. What was happening? So if we look at a couple of SST images, you see there is warm, salty water that has come off of the Gulf Stream that is actually moving up the up the shelf break. That water was lighter than the shelf water, so that reverses the density gradients across the shelf break. That's going to be driving flow to the north. If we look in the glider observations, this is now a mean again, like I showed before, but it's a mean only for that 3-month period of winter 2017-2018. If you look at salinity, the water along the shelf edge is very much warmer. There's none of that 34 PSU water. If we look at the shelf break flow, it is now to the north and pretty strong. So we've got 20 plus centimeters per second to the north, average over that period. So we had a complete reversal of the system during that period. There were only 2 transects during that entire period with any detectable export of Mid-Atlantic shelf waters at all. And coincidentally, there's an entire paper by one of Harvey's students on one of those events. There was a pretty interesting mechanism that happened, but that's for another story. All right, so I've told you this is episodic export, and I've told you the total integrated amount, but let's look a little more in detail at the spatial structure about that. And to do that, we'll look at eddy fluxes. For those that don't speak PO, eddy fluxes are just correlations of perturbations. So I'm going to I have a mean in a seasonal cycle. I'm going to subtract that away from all of my observations. That's what prime means. And I'm going to multiply 2 different perturbations together, and I'm going to average them. And that's going to give me an eddy flux of something. Now, one of the things I multiply in this case will always be the perturbation of the cross-shore velocity. That's the U prime. So here we're doing the cross-shore eddy fluxes of salt and temperature, which is more or less eddy heat flux, with caveats that those who know and care know what those caveats are. So what do we see? So the top panel here is the eddy salt flux. This is along the slope transect. So the Gulf Stream is at the southern end, 37 north on this side. And the lower panel is the temperature flux. So I mentioned before that you'd see this convergence line showing up. So that black line is where the mean along-shore currents go to 0. So north of that, the mean current is to the south. South of that, it's to the north. What do we see? So first, the biggest thing that sticks out to you is this big red blob signal down here. I want you to ignore that. That's the Gulf Stream. What is it doing? It is meandering laterally in and out of the frame. That is not something that's going to behave like what we think of as an eddy flux. It's not going to stir a gradient side to side. It is the gradient itself moving in and out of the picture. So ignore the biggest signal in this. Instead, focus on the negative terms. So a negative salt flux and a negative heat flux in this case are, I have to think and get it right, and it's too late to think. Negative salt flux is fresh water coming off of the shelf or salty water going onto the shelf, which is consistent with what we think of as stirring across the mean gradient that has warmer, saltier water offshore and colder, fresher water onshore. Ditto for temperature. So this eddy flux is what we expect for, or is adding heat and salt to the continental shelf, or removing fresh, cold water from the shelf. We see a near surface maximum in the salt flux near 36 north, right here. And then there's a broader and somewhat deeper maximum in both the temperature and the salinity fluxes north of that. So now if you permit a little bit of creative artwork, that might look a little bit like taking this cross-shelf structure and turning it sideways and exporting it offshore. So if you think the furthest inshore waters, if things aren't getting really crazy on the shelf, the furthest inshore waters are going to come out the furthest south as they make that left-hand turn to be exported. So this might be what we're seeing as an imprint in the eddy fluxes of the cross-shore structure. How about comparing the scales, or the quantity of the size of these eddy fluxes? If I integrate over the upper 200 meters, which is the part shown here, and I'm only going to integrate the negative part, so that ignores the Gulf Stream, what do we find out? So my numbers are here. These are from the gliders. Don't get too excited about the units. They are what they are. Comparatively, so Steve Lentz used historical records and inferred the eddy fluxes across the entire Mid-Atlantic Bight shelf break as a residual of what he could do for the budgets on the shelf. He got numbers that were a bit smaller, about half to 2/3 of what I'm getting. Glenn Gorkowitz from just some data from one cruise, so short-term data, more difficult to get a good average. But these were data south of New England, likewise got smaller heat and salt fluxes at the shelf break. So what do we have then? So the eddy fluxes of salt and heat across the shelf break are larger in the southern MAB, so south of 37 north. So it's a hotspot for eddy-driven, and here eddy doesn't necessarily mean spinny things, it just means time varying, but eddy-driven exchange between the shelf and the deep ocean. So one more piece, so that was eddy fluxes of heat and salt. We can also look at the eddy momentum and density fluxes. This tells us a bit about the underlying dynamics. So the momentum flux, that's going to be the U prime, V prime. That's the plot here on the top. And we again, ignore this big bullseye of the Gulf Stream. Everywhere else, that's pretty small. So U prime, V prime is typically used to consider baroclinic energy conversion. It's going to be U prime, V prime times the cross-shore gradient of the along stream velocity. If that's 0, that suggests a lack of barotropic instability happening in the shelf break front. I think we kind of expect that, but it's nice to see it borne out in observations where we can calculate the eddy fluxes. On the other hand, if we look at the eddy density flux, there's a shoreward eddy density flux, particularly further south. Baroclinic energy conversion, so baroclinic, which drives the baroclinic instability if it's taking energy from the large scale and putting it into the eddy field. It involves U prime density prime. That's the thing we're plotting over here. With the important caveat that if this slope of the isopycnals has isopycnals getting shallow or offshore, then the sign of the eddy density flux we're getting here is consistent with the presence of baroclinic instability. That caveat's important because remember I told you that the mean density gradient up at 37 north was actually pretty close to 0. So that's an important caveat. But it does look indicative of there being some baroclinic instability driving a lot of this variability near Cape Hatteras that is in turn driving the cross-shore fluxes. And so I'll leave it there and just some basic summary being we had 2 years of measurements that really did resolve this exchange. That exchange was highly episodic. It looks like the southern Mid-Atlantic Bight is a hotspot for eddy, salt, and heat fluxes between the shelf and the deep ocean. And that's largely driven primarily a result of baroclinic instability of the shelf break front. So there's a couple of papers out there. Harvey's paper is not in press anymore. It's actually out. Then I had a paper in JGR, and in the world of good data practices, the data are out there and readily available, and they have a nice DOI. So if you use them, please cite them. And that's it.
Okay, for all his whining about staying up late, he ran significantly over time. So we will get your revenge by holding him up after the meeting and talking to him then. Thank you.
Who gets this next?
Dr. Mubert?
There you go.
Great. Well, can you hear me well? Well, good evening, everybody. I'll cut it very short so we can get back to schedule. First, I would like to acknowledge and thank for the invitation. It's really a pleasure for me to be here. I think I haven't been in a science-only meeting for probably 5 years, where we only take science into consideration. So it's great. And it's also great to be here and see many friends that I haven't seen in quite a long time. It's been very fantastic for me. So I'm briefly going to talk about western boundary currents and early life history of fish. You know, I think Ian gave a great introduction this morning to this subject. Most of what I will be talking here has addressed in some way. So I'll just skip and make it shorter for you all. We can go to the next talk. First, let me know where I come from and why I think it's important to be here. I come from all the way south of Brazil. Rio Grande is located almost about 32 degrees south, so much further south than what Brandini showed this morning in São Paulo. And we are very close to the Brazil current and also to the Brazil-Malvinas confluence. So this is a place where we really take interest in study. And also, we are situated in the mouth of one of the largest fresh coastal lagoons in South America, the Lagoa dos Patos, which makes insignificant fresh water into the local coastal system, creating some real small-scale features that are of importance. And I forgot to mention, of course, that we are close also to the Rio de la Plata estuary, which brings in a significant amount of fresh water into the system, and I will address some of that when I talk particularly of that region. So, well, I will not go into all the importance of western boundary currents. I think you all here know that quite well. I will just mention that they are very important for fish, for fisheries, for food production. A lot of important commercial species are associated to continental shelves bordering the western boundary current system. And also, I'd like to mention that the western boundary current systems have the deeper currents and also association with the mesopelagic fish, which are believed to be a very important new source of protein in the future. There's been recent studies evaluating the size of these stocks, and we practically don't know anything about this environment. So my presentation today, it's focused on informing you the result of a systematic review we conducted on the literature to evaluate the studies on ecoplankton ecology in western boundary currents. I will not go through the whole study. I picked up a few examples to try to summarize what we know, or at least what I got from the literature that we understand on some of these different western boundary currents. So if your study is not there, don't get mad at me. I just had to pick a few. So, and maybe some of them are outdated already. I saw some very new stuff this morning that I hadn't seen in the literature because we only reviewed it up to 2021. So we use what's called the PRISMA method. It's called the Preferred Reporting Items for Systematic Review and Meta-Analysis. We define an objective, we ask a question, and then we look at the sources that we're going to look for the information. After that, we decide a period. It is open in past, so we didn't set up like an initial time, and we went up to 2021. Then you define a search strategy. You know, what are the keywords you're going to use? How are you going to combine them? Which is very important because you can get very significant results depending on how you look at that. You then create exclusion criteria. For example, we had larvae in there, but then for some reason we had some, crustacean larvae came out, or some other larvae which are not related to fish. When we got to look at western boundary currents, for some reason, estuarine systems came out, although we were not looking particularly to that. So we do a test and we validate the method, and then we search the titles and abstracts and we do manual reviews. And then you end up finding other papers that didn't come out in the literature review, which are of importance. So you have to go back and analyze them. So you look at the output, you perform analysis, and then you write the results. So we're in stage of really looking at the output, and I would expect to get some good input from you if I'm missing things or not. So I'm just going to briefly present some of the results we got so far. There are certain, at the end, we found 57 studies in the literature relating fish larvae to western boundary currents. They're distributed among the different currents, more or less 15 studies. 15 studies in the Kuroshio, 13 studies in the Gulf Stream, 9 studies in the Gulf system, 8 studies in the Brazil current, and 14 studies in the Eastern Australia. There's a period distribution here. Most of the studies were concentrated between 2000 and 2009 and 2010 and 2019. It's really interesting that the way we looked at, we couldn't find anything before 1991. And, you know, we know there's all the MARMAP studies here in the East Coast of the United States. There's all studies that should come up. You know, we are missing something here. We have to look back in the data. 20 of those studies looked at a single species, like what's the influence for anchovy or for a tuna or something like that. And others looked at the assembly, I mean, the community of fish larvae. And then we only had 9 studies that were actually looking at physical, what I would call dynamic processes, either eddies, meanders, or rings. And most of the studies are merely descriptive physical oceanography. You know, like we got the fish larvae distribution and related to some salinity profile or temperature profile, maybe density, and we don't go further in the dynamics of it. So this is the setup where we got the results. So I'm going to go through very quickly now in different boundary systems just to show you what we know. So starting with the East Australian current, I mean, I don't know, maybe I should skip that after this morning, but it's interesting because one of the first studies that came out in 2001, I guess, not the first, but one that studies that looked at the broader scale picture, you know, relating the Eastern Australian current with the Tasmanian current, looking at the otoliths, you know, the small increments that Ian mentioned this morning. And they found here that there's a region of mixing, you know, between these currents, and actually that the currents create some retroflexion that will feed back the individuals into the population in order to maintain these stocks. And that these seasonal boundaries actually do change quite a lot, so that the mixing area can shrink and increase with time. The same, more or less the same setting can be seen in the composition of different fish larvae that are there. So there's a group of fish larvae that are clearly related to the Eastern Australian current, other group of larvae that are from this mixed zone, and then a southern group from the Tasmanian current. So that this scheme of separation in the large scale also influences the composition of the species that we are there. Well, and then we go into this more finer tuned scales in terms of looking at the dynamic influence of the eddies, which Ian, I guess, alluded this morning very well and showed more, I guess, more some examples that are much recent than the ones that I'm presenting here. But it's basically what I want to bring here to you is that we already know when we've discussed the importance of like the eddies on entrainment of coastal larvae, and also the importance of these eddies in the dynamics of the reproductive strategies of some species, like in this case of the blue mackerel. So we know that in this case, we have a setup from the boundary currents that influences composition and also the dynamic aspects of fish larvae. In the Kuroshio system, also there's the same idea that the large scale setting also influences, in this case, we have the Kuroshio in this side and the China Sea currents in this side, and that they also in some way influence the composition of the different species in the different areas. So here are the species that are related to the Kuroshio current, and here are the species that are inside of the China Sea. it's interesting that here we found also an example where the importance of the frontal region in the fronts in the western boundary current, enhancing the abundance and the quality of the larvae that are found there. In this case, in this study for anchovy larvae, Osaka looked at different transects along the Kuroshio, and he found that in this first top panel here, you have the intensity or the density of nauplii larvae, or nauplii, I mean copepod larvae, which are basically a food for fish larvae. In this here, he looked at the condition of this food, looking at the RNA-DNA ratio, and then here the abundance of these larvae, and see that around in the shadow, in the gray area, you have the front. So in the front, you always have higher number of larvae, and you assume that these larvae are in much better condition because the food are in better condition. So this is fairly important to know that the frontal system also benefits early life history of fish. And the same thing happens if you look at the structure of myctophids. So this is very important oceanic group. You can see that associated with the western area of the Kuroshio, you have a particular group of species, and then another group of species in the central part of the Kuroshio, and then in the eastern side, you have a completely different set of species. So within even the same western boundary system, you have separations on this group of fish, which are all fairly, in terms of biology, if we compare, with a coastal system, they're quite a complex and similar system, the myctophids, the lanternfish. So in the Agulhas current, we basically don't have too much information on the dynamics of it. We have very few studies. We have many studies dealing with actually coastal systems like the Algoa Bay, looking at the distribution of this species. It was interesting that I found this study from Stirdam where she uses nurdles as indicator of larval fish transport. First of all, I didn't know what nurdles was, so I had to look in the literature. Do you know what nurdles are? Is this, yes? Well, it's basic plastic, right? So apparently they had, this is a citizen science project, so they had observations of nurdles displacement in all these different points, and she actually calculated displacement on them, and they are good tracers of 3 minutes. I'm finishing, of buoys moving around. So there is some information from, particularly from Olivar, from Pilar, studying the eastern the Agulhas current and looking at the distribution of different fish. So in the Gulf Stream, we've heard a lot of this system. It's interesting. There are many studies by John Hare and Bob Cohen in the 90s, looking at the transport of larvae from the southeast United States coastal shelf to the northeast United States coastal shelf, where they say that some species that are spawned here, they end up in this place. So they here suggest that the Gulf Stream can perform like the triad that Ian mentioned this morning. You have entrainment, you have retention, and then you have maintenance, right? Using these warm core rings. So further, John Hare also put together this collection of paper looking at the different dynamic features along the U.S. coast, which were important to fish larvae, mechanisms that could be important in the transport of fish larvae. So there's some very interesting information here on the physical process that we're looking at. Going back to my backyard in Brazil current, we have suggested that we have actually the formation of eddies in this region here in the southern part of Brazil that can trap fish larvae and enhances and changes distribution pattern. And also you saw this morning, I guess, Brandini showed the distribution of some of these cruises in this area where you can see actually where is the mouse now? Oops. Where you can see the importance of the, so in red here, you actually have the core of the tropical waters from the Brazil current, and you have some lanternfish associated to this core. So when the core advances over the shelf, you have these lanternfish happening in the shelf too. So this suggests in this cross-shelf transport. So just some food for thought. Many things we're not observing correctly at the right time scales and spatial scales. For example, in the southern Brazil, you can see, maybe you cannot see, but you see, rings or small meanders spawning out of this region going in the ocean area, but you don't see them inshore. And probably because here we have a lot of fresh water. This red, this is a chlorophyll image. This red is probably not chlorophyll, it's suspended material coming out of the Rio de la Plata. So this lens of fresh water prevents us from seeing anything from the top. Matsuda in 1996 proposed a mechanism to explain the failure in the sardine fisheries in 74, 75 related to subsurface chlorophyll maximum, and where you have intrusion of South Atlantic central water in the shelf and producing a lot of production when it's above the 1% light saturation, and that this has not only a seasonal variability but has interannual variability, which can lead to the failure of this study. And then we have topography. I will not go into this last slide because I'm really running over time, but topography is a very important feature. Here you have kinetic energy dissipation, actually vertical energy dissipation, and you can see that even at 700 meters, you have a lot of things going up in the vertical, and we don't understand that. If we want to look at these mesopelagic species, which live up to 2,000 meters, we have to understand what's happening in that area. So what's next? Well, most of the studies so far have been purely descriptive. We need to advance process-oriented studies and evaluate also the role of trophic structure and transfer of material within the plankton community. We know that this is an important thing, and not too many studies have been done. And we need to look at smaller temporal spatial scales. We just had a talk showing all these episodic events in the Gulf current. This happens everywhere, and this is probably very important because of time scales that Ian mentioned this morning for fish larvae. And we need to probably have more modeling studies using those IBM individual-based models and numerical and conceptual models to move the things forward. Thank you very much, and I appreciate any comments and suggestions. We can talk later on because now we have to go to bed.
Any questions here for Dr. Wheelbear? Getting late. I've got one. So in that last example, right, you've got fresh water in the South Brazil, right? And we see here that the stratification associated with that tends to increase the effectiveness of that onshore flow. But at the same time, with the additional turbidity, might also suppress the production at the same time. Do you see anything that suggests that might be the case?
Yeah, that's interesting. But suppression of production, I don't think it's a case there because the water doesn't get all that turbid. We still have some light penetration, which can promote production in the upper layer. It may not be very deep, but it's still very productive. Yeah.
Okay, you can put that down because our next speaker is on the TV screen, and that's Neil Mallon from the University of New South Wales. He's talking about the Pax East Australian current, separation variable, and shelf chlorophyll.
Okay, hi everyone. Thanks for the opportunity to speak, and thanks for staying awake so late into the evening over there to see it. I will also try and be brief. I'm from the University of New South Wales, and I'm going to be talking a little bit about some work we've been doing on the relationship between the East Australian current separation and shelf chlorophyll. And this is work I've done along with my colleagues, Minya Rowan, Michael Hemming, and Amandine Schaefer. So I guess we're moving across all the way to the other side of the Pacific here. For the people at this meeting, I don't think we need to have a lot of background, but the East Australian current, or EAC, is the western boundary current of the South Pacific Ocean. And I suppose in comparing it to other western boundary currents, I always think of it as having are relatively small and shallow dredge compared to, say, the Agulhas where I'm from. And because of that, it's highly eddying. Mesoscale eddies really dominate the system. It's got a narrow shelf along its entire length, pretty much. And it's also shifting polewards over recent decades, and we've got some quite large warming trends as a result of that. The part of the EAC system I'm going to be talking about today is over here between about 31.5 and 34 south, and we call this the EAC separation zone. And this is where the coherent jet of the East Australian current kind of splits into these two eddying extensions, the southern extension and the eastern extension. And one of the nice things in this region is that the Integrated Marine Observing System in Australia for the last, I think it's now 15 years, has been running glider missions down the length of this coast across the separation zones. And this means we've got in total 50 plus glider missions. We used 29 of them in this study. And one thing to note for thinking about Robert Todd's talk earlier is here, instead of doing in a long shelf transect, the gliders do a kind of sawtooth zigzag down the shelf, which isn't quite as nice for doing cross-shelf fluxes, but gives us kind of a really nice random sample of the shelf, which happens quite a few times a year. And then we can combine this with satellite altimetry and SST for kind of the large scale context, and then HF radar for the surface current details. And so if you have a look at what this long glider program looks like over the years, on the left here, you can just see the number of profiles per little quarter-degree latitude band, colored by year. So it gives us quite nice coverage, especially in the middle of the separation zone here. And on the right, we just have the coverage of this HF radar system. If you think the separation zone runs here between 31.5 and 34 south, and we've got about 3 years of data on that. And so in starting off this work, one of the ideas was we've got a lot of data on the system, and now what are the first kind of things that we can take out of it that aren't purely physical oceanography, and getting a little bit more into what's controlling chlorophyll in the system. So the area of shelf that we're really interested in this work, we call the Hawkesbury Shelf, in the separation zone 31.5 to 34 south, running from the coast to 200 meters. And what we know about this is that it's productive. So Everett et al. In 2014 did some work using satellite chlorophyll and found that at times just this little part of the shelf in this white box here is responsible for about more than 20% of the shelf chlorophyll of the entire East Coast. It's also interesting because it sits right next to a large coastal population of about over 5 million people, and it supports the local fisheries, and it's being in the separation zone, it's very dynamic. So this example on the bottom left shows some of the mesoscale variability you can have. This is out of a modeling paper by David Gwither. And sometimes we can have the EAC jet separating very far north over here, and then we have a purely eddy-dominated system. And then sometimes the EAC jet can penetrate all the way down to 34 south, and then we have what we would think of as a very jet-dominated system. The other interesting thing, seeing as a lot of the work in this region has been done from satellite, is if we have a look at some glider profiles, these are from Amandine Schaefer's work a few years ago, it seems that there's a relatively common, this is chlorophyll fluorescence from a glider, they're relatively common subsurface chlorophyll maxima in this system. And the drivers of productivity on the Hawkesbury Shelf is something that's been thought about for a while. So the first kind of studies I know of talking about the enrichment mechanism is Oak and Middleton in 2001. And what they found was that the upwelling was unrelated to coastal winds and was driven by the interaction of the EAC jet with the shelf through bottom boundary layer and other processes as the shelf widens. So the idea is the EAC jet runs along the coast, that drives upwelling, and that nutrient gets advected onto this wider shelf of the Hawkesbury Shelf and then drives phytoplankton blooms. The next year, Manini Rowan and Jason Middleton looked at actually comparing the various upwelling mechanisms. And the thing that they found is, in addition to these current-driven upwelling mechanisms, there's also a separation-driven mechanism. When the EAC jet moves away from the coast, it basically pulls up a whole bunch more nutrients, and this is really important for maintaining the deep nutrient pool in the shelf. However, since then, we've learned a few more things about the system, and a lot of these early studies, if they use a model, it's a short model run from climatology, and if they use observations, it's often quite a short observational run looking at a specific dynamic. And we know now that the EAC separation is highly dynamic, and eddy encroachment is also an important process for driving cross-shelf fluxes here. So what happens if we look at the system using modern observations and also our updated understanding of the EAC system? And the main important thing that we've learned here is on the separation modes of the EAC jet. So a couple of years ago, Nina Rabat led a paper where they use satellite altimetry to decompose the separation of the East Australian current into these three modes. So these are kind of the three main EOF modes. I've just made schematics of them here. And the first mode is what we call the EAC mode. This is just the EAC jet roaring straight down the shelf and separating quite far south here. Then the second most common mode is the eddy mode, where you have the normal separation plus a single, usually cyclonic mesoscale eddy, resulting in a slightly more northern separation. For those in the northern hemisphere, remembering northern here is closer to the equator. And then the last mode is the dipole mode of separation, where we have these two counter-rotating mesoscale eddies, and this is associated with an even earlier EAC separation. So I've just plotted this here over satellite chlorophyll from 3 days in recent years. The reason why these 3 particular days are chosen is because this is when we have gliders on the shelf. You can see the tracks there in orange. And if we first think about the Oken-Middleton hypothesis of more EAC equals more upwelling equals more nutrients equals more phytoplankton on the shelf, that idea stands up pretty well looking at this picture. We have an EAC mode of currents. We have quite high chlorophyll in the shelf here in the EAC mode example. In the eddy mode, we have less EAC jet, medium amounts of chlorophyll in the shelf. And then in the dipole mode, where the EAC jet separates really far north, we don't have much chlorophyll in the shelf at all. So that's a big tick for that idea. But now, because we have gliders and radar, we can look at the system in a different way. So we can look below the surface. So as I said, the reason we chose these 3 examples was because this is when we have gliders and radar on the shelf this time. And so now we have the mesoscale situation plotted on the right here. This is geostrophic currents and satellite SST. But on the left, we have current magnitude and direction from radar as well. And you can see the position of the gliders here marked in gray and blue. So takeaway here is during the EAC mode, as we expect, we have these strong south winds currents on the shelf edge and weak currents on the shelf itself. In the EAC eddy mode, so remembering that's this middle mode here, we have kind of medium currents all over the shelf. The EAC does a slightly strange split on the shelf. And we have this large mesoscale eddy to the south here. Then in the eddy dipole mode, we have almost the polar opposite of the EAC mode situation, where we have weak currents on the shelf edge and then quite a strong coastal jet going to the southwest here. And you can see this is associated with the cyclonic eddy over here and this anticyclonic eddy over there. So now what we can do here is look below the surface. So we just take these blue transects from the 3 glider missions as close to the same latitude as possible, and we can plot those out and have a look at the depth structure. So this is density on the left for those 3 examples, chlorophyll in the middle, and then just the across-shelf average of those 2 quantities on the right. So in terms of stratification, we can see EAC mode, we have this really well-mixed shelf water column. The eddy mode, we have a sort of shallow stratification, and the dipole mode, we have a deep stratification. Now, the EAC mode, remember, had the most chlorophyll at the surface, and here you can see why. We've got this kind of surface-confined, very shallow chlorophyll max. It's pretty well mixed through the top 20 meters of the water column, and so that gives us quite a lot of surface expression, but actually quite a weak chlorophyll maximum if you look versus the other modes. Then in the eddy mode, we've got a less coherent chlorophyll signature, this shallow subsurface chlorophyll max, and this very shallow stratification. And then the dipole mode, which, if you remember, had almost 0 surface expression of chlorophyll, then has this deep chlorophyll max at around the same depth as the picnic line, and that's really strong and has this deep stratification all the way, and a deep subsurface chlorophyll max all the way along the shelf. So that's already a bit of an interesting picture, but remembering these are just 3 examples. These are 3 example sections from 3 missions. There's a lot that could be going on in terms of long shelf variability, et cetera, et cetera. So the question then is, does this pattern carry on in the same way if we look at it under kind of harsh conditions. So the first thing we can say is this coherent along the length of the shelf? And what we can do there to do that is, instead of just looking at a single section along the blue sections here, we just take the time mean of the entire glider mission. In this case, they're all about 2 weeks long, and get a time mean profile for chlorophyll temperature, salinity, density, etc., and see if we see that same pattern. And long story short, we do. The eddy dipole mode still has the strong deep chlorophyll max. If we look at the time mean, the EAC mode is much more straight up and down in terms of the chlorophyll profile, and then the eddy mode is somewhere in the middle. And in terms of density, the signal is the same as well. This well mixed water column associated with the EAC mode, and then much more stratification associated with the other two modes. So that seems to work pretty well. So what we're seeing from these sections seems to be mostly consistent along the length of the shelf from 31.5 to 34 south. However, we then get back to the thing, these are just 3 particular missions that we've chosen. What about over time? Is this the normal pattern for the EAC, eddy, and dipole modes of separation? And then, of course, we've got 29 glider missions, so we can then decompose each of those glider missions into whether they're in the eddy mode of separation, the dipole mode of separation, or the EAC mode of separation. And we can make composite means of those. And when we do that, we still see that same pattern in terms of density. The EAC mode has the most mixed mean profile. The eddy mode is much more stratified, and then the dipole mode is the most stratified. And in chlorophyll, we have that same signal. This dipole mode has this strong, deep chlorophyll max, and then the EAC mode is much more well mixed and much more straight up and down near the surface. This gets a little bit more complicated when we think about seasonality and include this. In this case, we've made the same kind of composite mean of the glider emissions, but by season. In this case, we've just divided into warm and cool seasons, with November to April being the warm season and May to October being the cool season. And we see a very similar pattern to what we saw dividing things by mode, where in the warm seasons, we have this deep, strong subsurface chlorophyll max, and in the cool seasons, things are much more surface intensified. So this is already interesting because this area from the surface has a normal spring bloom pattern where the phytoplankton, the chlorophyll, the surface peaks in August, September in the springtime. And now we're seeing at depth that is actually slightly out of phase and happens a few months later. So the seasonal cycle is out of phase at depth at what we see at the surface by a couple of months. And then there's this relationship between the EAC separation and the seasonality. And if you look at the individual missions, which we can talk about later if you want, you actually see that the dipole mode has a really large seasonal range in its chlorophyll. So if you get a dipole in summer, you get a really strong chlorophyll response, in winter, not so much. Whereas, for example, the EAC mode shows no clear seasonality. So this seasonality here is also dependent on what separation mode we have. And then we start thinking about what drives the dipole mode being so productive compared to the other two modes. And one of the first things we think of there is cross-shelf flow and cross-shelf exchange. And so for these 3 examples that we've been looking at, we can estimate this, not particularly accurately, but it gives us a good idea from satellite altimetry, so just geostrophic velocities across this red section. And if we look at our EAC mode case study, as we'd expect, we don't have a lot of cross-shelf flow in the northern half of the domain, and then we have the southern poleward separation of the current down here. That's as we expect. In the eddy mode, we have this cyclonic eddy and an earlier jet separation, and you can see this in the cross-shelf velocities, far more distributed towards the north of the domain. And then in the dipole, we have that same northern separation, but here we have the onshore transport that is contributed by this counter-rotating dipole that directs a jet of offshore water onshore. And then we can ask ourselves, does that really matter? And we've done a little bit of work on this in the past, both from cruise data as well as modeling. And we've shown that we think it probably really does matter. From shipboard ADCP, we showed that the transport of this dipole jet between the 2 eddies is about 16 svadrup, which, considering the transport of the EAC is about 35 svadrup, is quite a significant number. It's almost 50% of the total flow of the EAC. And when we do this much more accurately and look at cross-shelf transport across the 1,000-meter isobath from a ocean model, a high-resolution ocean model, we can see here we get around the same numbers. We get 12 sphagips in the snapshot, which is April Fool's Day 2009. And at the time, this is the strongest single cross-shelf flow, cross-shelf transport along the length of the whole EAC system. So it appears the dipole has the strongest stratification. We think that's related to this cross-shelf transport, and that seems to be the mechanism by which we're getting this increased deep chlorophyll max, which is quite productive. And this is something we've also seen before a few years ago, where we did a bit of a deep dive using this cruise data into what was going on during this dipole. And the main thing is you have this kind of really strong deep stratification on the shelf. And you can see here, this is from a glider, by the way, that we have this deep chlorophyll max above it. And it seems like there are a whole bunch of processes once you start stirring with eddies, the shelf water column. That means we get a transfer of nutrients across a kind of deep picnic line that then results in this deep chlorophyll max. And I think that's been observed in other parts of the ocean, just usually the straining, stirring mechanism there is tidal rather than being, in this case, by mesoscale. That's still a little bit conjecture at this stage. So just to summarize, what we found in this system, subsurface chlorophyll maxima are common, much more common than we initially thought. From all of these glide emissions, I think we can say that a more stratified shelf tends to be more productive. And this is probably to do with some eddy-driven stirring mechanism moving nutrients across the picnic line. The seasonal cycle is out of phase at depth with that at the surface, but that is dependent on what separation mode is dominant in that particular season. And then another thing, and I think we spoke about this earlier, and there's a session on this later in the conference that we need to think about, is what are the implications of these kind of mechanisms, particularly the dipole on the physical and biological carbon pump. If these are hotspots of CO2 uptake, here in the dipole, we have a mechanism that causes a lot of productivity quite deep in the water column at the same time as a really strong cross-shelf exchange mechanism. So what does that mean for carbon export, and also how accurately do we need to model these systems if we're going to do, as suggested earlier, and try and get a budget of how much carbon is being exported globally? I think we're going to need simulations that can kind of accurately simulate these eddy-driven processes, because they're an important component. That's it for me. Thank you very much.
Anyone with a question? It's up to me again. Okay, Neil. I mean, you've obviously shown a deep chlorophyll maximum, but you're talking about production, and they're not the same thing. So, do you actually have independent measures of production, and not just an overexpression of chlorophyll in cells?
Yeah, well, that's a classic. Robert was saying earlier about us physical oceanographers should stay away from the squishy stuff. So yeah, that's me using wrong terms. We're talking about chlorophyll here. We haven't got production numbers at this stage.
Use oxygen.
Yeah, I really didn't understand how you get the subsurface chlorophyll maximum. Perhaps you can clarify it to me, because what I see is that with the dipole, you have inflow of water onto the shelf. These waters came from the surface layers, which are nutrient depleted. So how you get the subsurface chlorophyll maximum?
Yeah, that confused me a lot earlier in the early stages looking at this data, that theoretically what should be the most unproductive mode, when you think about it, with this unproductive offshore water moving onto the shelf, turns out to be the most productive. And I think that comes into this increase in stratification that is then creating an environment for the subsurface salinity maxima of increased nutrients that's in that deep nutrient pool. But I have to say, we don't fully understand that.
Yeah, hi, Neil. Great talk. It came across really well all the way from Australia. I just wanted to congratulate you, but also to remind us all that where you do see a high biomass, it's usually, not always, correlated with a high production. So that squishy stuff is being delivered into the Stockton Bight, which is a really productive area for lobster, for fisheries. And so this mechanism and so trying to quantify the chlorophyll budget onto the shelf for fisheries production and how that changes in the future is really important. So I think it'd be nice to go and follow this through, to integrate it over all the the years that you can assess this sort of dipole mechanism to see its contribution. Can you do that?
Oh, sorry, and I just lost you for a second there. Can you just repeat the last part of the question, please?
Just the last part was, can you estimate this dipole production mechanism onto the shelf, into the Stockton Bight, over the past 20 years, just using altimetry, looking at the dipole mechanism, and how much chlorophyll does it contribute to the overall New South Wales shelf budget?
I guess I don't know is the answer to that question. We could look at it. We do have a couple biogeochemical model of the system, but we're not 100% confident in its performance on the shelf. Altimetry, you can get estimates of cross-shelf exchange. We can do the same using the model, but that missing link, I think, is, how do we get from cross-shelf exchange, first of all, to biomass, and then to production, and then to export? And I think there's quite a lot of work to do still there.
Okay, thanks.
Do you have data on carbon to chlorophyll information, carbon to chlorophyll ratio, in such a way that if you have chlorophyll stocks in that particular place, with that particular area, you can have also the carbon stocks, carbon from the phytoplankton, of course, not the total carbon. Or usually out of the coast, when you don't have too much interference from detritus, most of the carbon comes from the water column itself. So if you have a fairly good ratio of carbon to chlorophyll, with the chlorophyll data, you can make a very good modeling on carbon production due to your dipole, not using chlorophyll. Chlorophyll is not biomass, it's just an indication of abundance, sometimes not even that, because some species have more chlorophyll because they photoadapt. It doesn't mean more carbon. But if you have data on carbon to chlorophyll ratio, then you can start a new approach in terms of biological pump, carbon pump, what your dipole does with this, right?
That's a really great thought, thanks. And that's something, there are some studies on kind of those kind of dynamics, but they usually only, it's a short-term study looking at one mode of the current, but that sounds like a good way of extending that to the greater system and its full variability.
Thank you, Neil. And let's thank all the speakers for their efforts tonight. Tomorrow morning, we'll have coffee again down here at 8, starting at 8, and then pick your poison upstairs after that at 9 o'clock.